and they are actually susceptible to any abiotic changes. So as I previously said, our today's species is Rhizopladus olivacea, which is the most common Heaton species in the Eastern Mediterranean Sea, mostly in Greece and Italy. And they can be found in depths of 0.5 to 62 meters. So the autapomorphy of mollusks is the radula, which is a specialized feeding apparatus consisting of hundreds of mineralized teeth rows that they actually have varying concentrations of magnetite and apatite, which makes it the hardest and stiffest biomaterial on Earth. So in general, there are 7 trophic gills with specialized radula. So we actually, for this study, use the relative literature to categorize our radula into 4 different tooth types, namely central, lateral 1, and lateral 2 and marginal tooth. As you can see in our photo by the different colored circles. And lastly, there are no studies, actually there are no data on the specific variation in the Heaton mineralogy on the radula. So in total, there were 50 individuals collected in July 2023, and using scuba diving in depths of 0.5 to 2 meters. And they were transported alive to the laboratory for processing. So we chose 3 sampling sites in the Eastern Mediterranean Sea. In the North Aegean, the sampling occurred on the Chalkidiki Peninsula, while on the Central Aegean, we chose Pagasetikos Gulf. And lastly, in the Ionian Sea, the sampling occurred on Patsos Island. So, to examine the mineralogy of the radula, we firstly dissected all the individuals to acquire the radula. After that, we actually rinsed with distilled water the radula, then dried it, and lastly, coated in carbon. After all that, as we prepared all the samples, they were actually analyzed and displayed by SEM, followed by EDS analysis in order for us to acquire the spectra of the radular mineralogy. So this statistical analysis of the radular chemical composition, we saw that the highest mineral concentrations were iron and calcium elements. Firstly, in the lateral 2 tooth, as it is the biggest teeth in the radula, there were the lowest calcium concentrations and the highest iron concentrations. Meanwhile, in the marginal tooth, which is on the outside layer of the radula, we had the lowest iron concentration. And as for the central lateral 1 tooth, it was examined that they had both similar patterns as for the iron and calcium concentration. So, as you can see at the figure of the PCA analysis, iron was the most important variable at all tooth types, while calcium, magnesium, and phosphorus were located opposite to iron. So as for the regional profile of the Rhizopladus olivacea radular mineralogy, we can see that the Pagasetikos Gulf was differentiated from Patsos Island. As for the central and lateral 1 tooth, continuing to the PCA C figure, we can see that the lateral 2 tooth of Chalkidiki was differentiated from the other 2 sides. And lastly, the marginal tooth, all 3 sides differentiated from each other. So in our discussion, as we saw at the figure of the previous slide, there was a clear pattern of intraspecific variation in 3 sites, each with different environmental factors. So in general, the main theory behind the radula mineralogy is that the chiton, by scraping of the rocks algae, it's actually wearing the toothing down, which forces the chiton to spend the energy in order to renew the radula. In order to cope with the wearing, the chiton is actually increasing the hardness of the radula, in which the environmental factors might have a major role, as we can see in our study. And lastly, the increased hardness on the radula is actually inhibiting the wearing of the radula, which helps the chiton save some energy. So what is our future goal for this study? Our future goal is to continue examining the Rhizoplagiolivacea mineralogy in order for us to determine if the chemical composition as well as the mechanical factors of the radula works as an adaptive trait in order for these species to occupy not only different geographic regions, but also different ecoregions. I'd like to thank you for your time and your attention. The first region by Stikos Gulf is where our department was established. And after that, we actually tried to find any other sites of the Rhizoplagiolivacea. And we actually tried to find in the different ecoregions like North Aegean, Ionian Sea, South Aegean, but we actually find only in the Chalkidiki, in the places that we actually tried to search for the species. And then the Ionian Sea, it was a Paxos Island.
What are the main environmental factors that can save this distribution of the species?
I'm talking about factors such as temperature, salinity, pH values, even if the different topography of the environmental might play as a factor. Thank you very much.
Thank you, Mr. Migalia. So moving on to the next presentation by Mr. Zappos. It's going to talk about short-term effects of extreme storms on population characteristics of the continent of the MMT.
Greetings to everyone. My name is Zachos Daniel Solnas, and I'm a graduate of the Department of Ichthyology and Aquatic Environment from the University of Thessaly. Today, I will present to you the short-term effect of extreme storms of a population characteristic of a common Mediterranean chiton. On 8th of September 2023, Daniel, an extreme rainfall event occurred in Thessaly, followed by a second extreme rainfall named Elias 20 days after. Pagassitikos Gulf was the main recipient. Photographic evidence provided in this presentation showcased the aftermath of the two extreme storms. We can see in the two upper photos the local area of Agios Stefanos before and after the two catastrophic rainfalls, while In the two bottom photos, we can see the area of plaques before and after the catastrophic events. The two extreme rainfalls affected the local intertidal and shallow subtidal organisms. One of those organisms is Polyplacophora or Chitons. Chitons are among those affected. They are considered important grazers, while they have also been characterized as bioeroders. They are exclusively marine organisms, and the majority of Chitons are xenohaline. Unfortunately, Polyplacophora is an understanded taxon in Mediterranean Sea, so we have limited knowledge about them. Finally, they are severely susceptible to abiotic changes as their growth, reproduction, and feeding are highly affected. Marine coastal ecosystems are very vulnerable to climate change. The two extreme storms that occurred in Thessaly are a result of said climate change. The three species that can be found in the studied areas are namely Acanthochitona fascicularis, Rhizoplax olivacea, and Lepidopterus cajetanus, with Rhizoplax olivacea being the most common in Eastern Mediterranean Sea. After observing the boulders in the studied areas, we found out that the previously abundant species boulders were now unoxidized and poor of life. So the ultimate goal of the study was to estimate the effect of extreme storms on density, growth, reproduction, and feeding of Rhizoplax olivacea. Some collection was carried out during October, November, and December, and was repeated after the storms 1 year later, the same months, with the ultimate goal of comparing them. For the sample collection, we scuba dived in 0.5 meter to 2 meters depth in 5 replicate quadrat plots. After the sample collection in the laboratory, a relaxation protocol was followed, which consisted of gradual dilution of seawater to freshwater, while simultaneously adding drops of 10% ethanol. This protocol was used to measure the full morphometrics of Chitons because Chitons have the ability to contract under stress. Finally, we dissected the Chitons to measure the gonad weight, radula length, intestine length, and food weight. The densities were measured by counting individuals of index quadrat, while 6 indices were calculated to estimated reproductive energy and somatic status, those being namely gonosomatic index for reproduction, relatively radula and intestine length, and elongation index, food somatic index, and body mass index. Abundance of Rhizoplax olivacea was declined in both sites, but while a gradual decrease in density was observed in Agios Stephanos area, in Plaques, and almost sudden absence occurred, as the line plot suggests. Individuals inherited a more elongated body shape after the two catastrophic rainfalls, while both gonosomatic and food somatic index decreased. Also, body mass index was decreased after the events. Finally, trophic indices were exhibited higher values after the two extreme storms. The gradual decrease in density in the western part of the Gulf in comparison to the instant mass mortality of the eastern part could be due to possible migration because of the environmental stress. The elongated body shape could be an adaptive behavior to the increased wave action. It could also be a sign of natural selection. The increased body mass index possibly occurred as an energy conservation mechanism to cope with increased stress, while the decreased gonosomatic and food somatic index evidently shows that the reproduction might be hindered and the growth might be stagnated. Finally, the increased relative intestine radula index might be associated with food scarcity. It appears that the two storms impacted the ecological aspects of Rhizoplax olivacea in Pagasetikos Gulf. However, these changes are only describing the short-term effects thus encouraging the long-term monitoring in order to assess the potential recovery and adaptation of these species. This knowledge will serve as a further step to identify patterns of adaptation of chitons and mollusks in general in a changing climate. I would like to thank each part of our team, those being Kostas Vulgaris, Varkoulis Anastasios, Tomas Migdalias, and Dimitrios Vafidis. And I would like to also thank you for your attention.
Very much for the very interesting presentation. Are there any questions? No? Thank you again. So
Thank you very much.
Good one from the beginning of what I say this. Let's hear about that. Okay. Dr. Zakharov, give us a presentation about turtles, about the preliminary results regarding turtles nesting at Italia.
Yes, yes. Yes, it's okay. Hello, everyone. Allow me briefly to introduce myself. I'm Ertan Taşkavak, coming from Turkey, Izmir, working at Ege University Faculty of Fisheries. I'm here to talk about the significance of Manavgat coast in Antalya region. So our observation based on over 3 years, mostly numerical data, yes, or elevated data of 2022. As you know well, two sea turtle species use Mediterranean coast for nesting and egg laying. What are those species? Those are caretta caretta loggerhead turtles and Cheloniamidas green turtles. Except for those two turtles, there is also leatherback turtles, Dermacoelis coriacea, seen very often in Mediterranean as well. Caretta caretta, the loggerhead turtle, primarily nest in Greece, Turkey, and Cyprus, and Libya. A little bit nest was discovered in Libya as well. While the green turtle's main nesting area are in Turkey, Cyprus, with additional nesting records from Lebanon and Israel. Okay. The leatherback turtle's Dermacoelis coriacea is a species that is regularly seen in Mediterranean. The husk-billed turtle Erythromacheles imbricata and Kemp's Ridley turtle Lepidochelys kempi occasionally make appearance, typically in Eastern Mediterranean, about Spain or France coastline. The IFCN listed Cheloniamidas as endangered, Caretta caretta as vulnerable. We primarily focused our research on high nesting areas such as Dalian, as seen in picture, Fethiye, Patara, and other newly discovered beaches. Currently, the government identified 21 beaches as sea turtle nesting areas, which are marked on the map, as you see. These beaches extend from Ekincik from the west to Samandağ, in the eastern side of Mediterranean. All those beaches are under the protection by relevant ministry. The map reveals that some beaches hold primary importance, while others are of secondary importance. A few remain unsustainable or unregistered, indicate in italics. Moreover, we have observed nesting processes at unofficial beaches such as Alanya and Erdemli and the Manavgat beaches, but those 3 beaches are not seen in the map because it is not registered by the government yet. In this what I'm going to talk about in this presentation. In this presentation, we will explore turtle nesting activities and challenges on Side, Sorgun, and Boğaz unregistered beaches in Manavgat coastline. Manavgat coast cover a coastal strip from Manavgat River on the east. You see the Boğaz. The Manavgat River's breakwater or pier in the east to Side's ancient city in the west. These beaches, stretching approximately 10 kilometers and ranging 50 to 100 meters wide, slope gently into the sea and consist of fine, coarse, grained sand. So, Manavgat coast is bordered by the Manavgat River and Sides Cliff at the west. Next, you see the Boğaz on the left side. Manavgat coast is located in the same coastal band as Belek and Kızılot. You see a red spot on Manavgat. In west there is a Belek, in east there is a Kızılot. Both are under the protection of relevant ministry. It's a narrow beach nestled between the river and the sea. The river is Manavgat River, which is the famous sightseeing area in Antalya region. And adjacent to hotel regions. For monitoring, we divided these beaches into 4 distinct areas based on human activities. The first area in the private-public beaches, which is primarily used during the day. Nighttime activity is forbidden there, with the minimal activities illegally passing at night. This area is located on the Side side. You see right side at the bottom picture, Side side. Before the construction of the breakwater or pier at Manavgat River in the 1980s, this was followed by the building of the pier in early 1990s. Finally, the 2 photos describe the current state of the beaches as of 2024. I took the left side and right side pictures before coming to here. The second area is the hotel zone. Those hotels differ in their distance from the beach and their respect for the sea turtles and the work we try to do there. The third area is the Manavgat Municipality public beach and vehicle road. You see on the right side bottom, which stays illuminated throughout the night. Lastly, the 4 is the Boas breakwater coast. It is home for private public beaches, mainly used during the daytime. So, in brief, you can read the segment of those 3 beaches, Side, Sorgun, and Boas, along the 10-kilometer stretch of Manavgat nesting and egg-laying coast on Google Map. Material method, what we do during this project. In 2022 breeding season, we followed global monitoring protocols. Every morning we marked nest and non-nesting sites on the beach. We studied the females and their distinct tracks on the sand. Using a metal stick, we separated nest from non-nest. We traced tracks from each nest, recording hatchling reaching to the sea. After all hatchling emerge, usually 3 to 5 days after the final emerge, we examined the nest. We counted empty shells, infertile eggs, dead embryos, and living and deceased hatchling. We set up a wooden signs at all nesting sites for protection and public awareness. What is the breeding activity in Side, Sorgun, and Boas, let's say Manavgat coast? Out of 330 turtle sightings at Side, Sorgun, Boas beaches, 250 resulted in nest. This means 75.8% of all emerges. All 3 were green, only 3 were green turtles, while the rest were loggerhead turtles. The most active areas were the private public beaches on Side side and Eastern Municipality Public Beach and the breakwater area between the Manavgat River and the sea, especially in the areas with no vehicle access and daytime only private beaches. It is seen in the map. Okay. Private day-use beaches and vehicle-free beaches have the most nests. All 2022 studies revealed turtle activity, both nesting and otherwise, occurs from June to July. In May, we have a very few, 5 or 8 nests. Last year, in 2002, last year was also same, but this year, before coming here, in May, we have 75 successful nests, and 2 of them were green turtle. Okay, when examining the nest density distribution on the beach, nests are more dense in the areas with less nighttime human activity and poor lighting. We observed more nests in hotel distance central areas, peaking at 107 nests in public beach breakwater area. The beach between the river and the sea in the public beach breakwater area peaked with 107 nests. Okay, let's talk about a little bit numbers. During the 2022 breeding period from May to August, we observed 5 nests in May, 126 in June, 93 in July, and 16 in August. If we consider total emerges, including both nesting and non-nesting, 2.4% were in May, 55.8% in June, and 37% in July, and 4% in August. In the study area, we found 35.9 emerges per kilometers and 27.5 nests per kilometers. The West Shore Band measures between 50 centimeters to 5 meters with an average of 1.95 meters. Loggerhead turtles had incubation period of 44 to 65 days, which is very short time for expected incubation period, averaging 54.9, let's say 55 days incubation period for loggerhead turtle, while green turtles average 57 to 58. It is very close to expected incubation period for green turtles. Okay, the Manavgat coastline, home to hotels, beaches, and picnic areas, is suffering degradation due to construction activities conducting comprehensive biodiversity surveys before approving future project is crucial to prevent further damage. For example, sand lilies, once abundant before the surge in coastal infrastructure, have seen a significant decline. A lack of research, however, hinders our understanding of impact on various species. This area is sympatric usage of caretta, Chelonia midas, Trionyx stringuis, monk seal, monachus, and also sand lilies. All of them are protected under the IUCN list. The breeding season of sea turtles coincides with the increased human mobility due to summer tourism. Of course, this often has detrimental effects. However, looking at the positive aspects, many hotels have implemented eco-friendly practice, such as these includes maintaining clean beaches, avoiding nighttime activities on the beach, promoting green tourism, and restriction vehicle use on the beach. Despite these efforts, all observations indicate that Boğaz Public Beach, which is under the control of municipality of Manavgat, poses a greatest threat to both sand lilies and sea turtles. Many construction in the region overlooked environmental implication, leading to considerable human activity in that region. You see the sand lilies and the garbage on the beach. Human activities, including littering, water sports, and incorrect fishing, contribute to turtle deaths. It is a well-known fact that artificial lice misguide hatchling. To mitigate this, these lice have been replaced with dim red ones. Silver gulls, carrion crows, and feral dogs pose threats especially to hatchlings in Manavgat. In 2004 study, Can Bolat found that 80% of Mediterranean turtle nesting occurs in Turkey. The Manavgat coast hosts about 250 to 400 nests annually. It's approximately 8 to 23% of total nesting in Turkey. Side Sorgun Boğaz beaches have more nests per unit distance, indicated that they are more productive than most of the beaches under the protection in Turkey. Although nest numbers in the Mediterranean have increased by 47% due to the conservation efforts based on about 4 decades, the lack of protection and monitoring on Side Sorgun Boğaz beaches present difficulties for comparison because it's a new discovered beaches. We partially persuaded the hotels to reform, to change their attitudes against the beach and against the guests what they have. However, due to election tactics in 2023 last years, we couldn't influence the Manavgat municipality. The adverse condition on the unregistered Side Sorgan Boğaz beach may impact both legally protected Kızılok beach as well as the Belek beach to west. So, consequently, our objective is to gain official protection to Side Sorgan Boğaz beaches and increase public awareness about the urgent matter. We plan to boost scientific monitoring and preservation efforts with the aid of volunteers. I will speak now. It's equally crucial to promote species conservation through our teamwork and educational activities among the stakeholders, starting a dialogue with local government, tourism facilities, law enforcement agencies, local business, and the school is a key step toward achieving our goals in Manavgat beaches. Thank you for your participation and patience. I tried to visit the centers in Greece, but it was impossible. It was impossible. Thank you very much. All these observation and monitoring programs carried out with the volunteers, with the young people like you. So it is under the umbrella of DECAFOC, and non-governmental organization. So I'm also the member of this non-governmental organization fighting against the state, municipality, as well as the government in Ankara to protect those animals and also environment. You can be the member of this organization, and also you can be the volunteer for this year or for next years by just sending an email to the chief of the NGO DECAFOC. Accommodation, I mean lunch, breakfast and dinner is free of charge, as well as the hotel free. Thank you very much. Yeah.
Actually get some measures against degradation, but they didn't follow them, they were unsuccessful.
I'm sorry, I couldn't understand what you mean.
You said, I think that the hotels took some measures.
Yes. We persuaded them one by one, visiting the manager of the hotels, and we gave the lectures how to protect those animals. And not only the guests, also before the season, we compiled the workers in the hotels with the permission of the hotel's owner as well. So we gave the presentation to them, also to staff. So they are much more sensitive against the nature, against the animals. For example, before they don't let us to put a sign on the beach where the tourists making sunbathing, but now it's okay.
But the problem was when you solved in the future.
Oh no, it's not easy to say so simply, because one side is a hotel, the other side is the municipality. Municipality itself make a very leisure area to improve the election, to improve the chance of to be elected, let's say. So all nights they used a very light lightning at the beach in the Boğaz area, where the nesting and hatchlings made peak in that area. We try to, yes. We try to persuade the municipality, but they say no. Also, this area is used nighttime as well for court-shipping behavior among the Homo sapiens, because it's a remote area. Thank you. Okay, thank you very much.
Okay. Okay. Okay, good morning, everybody from me. My name is Vasilis Papathanassiou, and I'm going to present you something about combining different methodologies for mapping seagrass beds. I'm sure most of you know what seagrasses are, but just in case, they are not algae, they're not seaweeds, they are aquatic plants. They have real roots and they produce flower and seeds. They are considered as to be one of the most important coastal ecosystems that we have, habitats, due to the numerous ecosystem services they provide to us, such as the ones that you see here. I will not go into detail. Like most things on our planet, they are threatened by us from numerous pressures. I'm just highlighting here three: fisheries, especially bottom trawling, agriculture, and tourism, meaning coastal development, eutrophication, and small boat anchoring. As a result, 30% of those meadows have been lost globally. The species I am focusing here is Posidonia oceanica. It's the endemic Mediterranean Neptune's grass. It grows from 0 to 40 meters, and you can find it almost anywhere. Even for a seagrass, is a very special species due to the high biodiversity that it hosts, as well as the very long periods that it stays alive, from hundreds to thousands of years, making it one of the most important carbon storages that we have. And of course, we protect it through different conventions, directives, and regulations. And a lot of work is being done towards that end. And something that came as a surprise to most marine ecologists and biologists is that when you try to protect something, it's not enough to know the species itself. You need to know exactly where it is. So here comes the need for proper and accurate maps. And when we are talking about mapping, you have different kinds of protocols, but the most common ones are acoustic methodologies, where you're using basically sonars that transmit sound and you get to scan the sea bottom. And this is a great way to achieve high accuracy, but it is dependent from the hardware that you're using, the research vessel that you're using, the depth you are focusing on. So and it takes a lot of time. So all of these translate into a high cost. So you have great accuracy, but at high cost. On the other hand, you have remote sensing, which is gaining nowadays. It's fairly fast, way cheaper. Using all these new computing capabilities, it's very easy to achieve a habitat mapping with less time spent in the field. However, since we are talking about the sea bottom, the overlaying water column significantly affects the attributes, the physical attributes of light, so you get a lower accuracy result. What we tried to do is change to the next slide, which is not working. Nope. I'm sorry here, guys. Let me do it this way. Waiting for it to get into full screen. So basically, this was our train of thought. So we thought that originally we got a Sentinel-2 image, which is free. You can get it. I don't know if you want to have a go. You can get it for free. We used that product. We created a first mapping of our study area, which was Samothraki Island in the North Aegean Sea. And we did that with no ground truthing data, just by using what was available in published information and ecological knowledge. And we did that just to get an initial map to get our bearings. So supposedly we have no idea about the area that we are visiting. We wanted something. No, it's okay. Okay. Then using that map and again published information, we identified some areas of interest. And those areas of interest, we wanted to focus more specifically and get more detailed information. In those areas, we then visited and we mapped them using a side scan sonar, the one that you saw previously in the picture. So at this point, we have some areas of from the complete region that we wanted to map using a side scan sonar, and then we got a second data set with points, with optical data using underwater cameras and video. So these two data sets, one from the side scan sonar and one from ground truthing points, we used to reach to our final product. Is it okay? Okay. This is Samothraki Island, and these are the limits of the Natura zone. We are focusing on the Natura zone there, and this all work is being done within the implementation context of the Marine Strategy Framework Directive. So this is the initial map I was talking about. This is a product, a map that we created without using any ground truthing points, and that is irregular. That is not a proper way to go, but keep in mind, we didn't do this to use it. We did this just to get a feeling of the area. Supposedly, we wanted to develop this methodology about going to an area that you haven't visited previously. This is the field survey plan that we designed. Now, you have two things here. You have those lines that they represent areas that we wanted to scan using the side scan sonar. You have one line parallel to the coast, and that is very important because you really need to have an accurate map of the lower limit of Posidonia oceanica, which is an important characteristic of the habitat. And you have all those points scattered within the Natura zone. Those are points that we went with a camera and we got our data. These points are more dense in lines that you see a side scan sonar, and that is because sonar methodologies also require a proper ground truthing survey. And of course, we had some scuba dives. This is how Posidonia looks in side scan sonar, those puffy, cloudy images that you see on the top. Those are the Posidonia patches. And if, I don't know if you can see it, you can also see the sand dunes in between. So you have a really accurate image of what's going on. You take all that data, you use a specific software, you combine them, you create a photo mosaic. You take that mosaic, you put it in a GIS, you start digitizing these patches, and you end up with a layer that has polygons of Posidonia oceanica, and this is your first data set. And then you have all those points that I told you earlier, and each one of those points goes together with an image which is geo-referenced, looks something like that, depending on how you got it. You end up with a second data set, and those two data sets are the ones that you finally use in order to retrain your algorithm and reanalyze your satellite image, and you end up with a final product that looks a bit like this. In our area, you can see that it is way different than the first map I showed you. Of course, this is expected. Here, I have used a lot of really good, accurate ground truthing data. You see that the meadow is close to 5 square kilometers. It reached to 23 meters at the deepest part. However, most areas, the meadow finished at 16 to 19 meters. And that's not a good sign for Posidonia oceanica. Usually it reaches at least 20 meters, even in the Northern Aegean, which is a very turbid sea. However, we attributed that to the really strong currents that are present in that depth zone. Also, you see some breaks, and those at the beginning, we didn't know what to make of it, especially one big break on the meadow that you see at the northern east. And but then we started looking at what's happening in the shore, and that is an area that there's a lot of freshwater inputs in the sea, and Posidonia just doesn't tolerate fresh water. It doesn't like it. So probably that is attributed to those waters there. So this is what we came up with. So was it worth it? This is the original map I showed you. This is a case that you just want to do very quickly, a map from your office using only published information. And this is something that happens. A lot of people do this. This is the product that we ended up. It's not bad in terms of accuracy. You get that 0.71, depending what you want, you can tolerate with it. This is what happens if I only use the ground truthing points to train my algorithm. And this is the proper methodology that somebody would use if they wouldn't have a side scan sonar. So here you see the second layer. It's closer to the final one that we get, so especially towards the limits, but there was a problem that you probably cannot see here. The classification gave up a very sparse meadow, and this is why you do not see a significant increase in area. However, accuracy is increased as expected. And finally, if we put the side scan sonar data in, you get a far better result, which is quite close to the truth. I mean, we've been there. It's very important to see an area with your own two eyes. And accuracy is quite acceptable. It's quite good. 0. I don't know if any remote sensing people are in here. So just some things to keep in mind. Remote sensing is crucial for monitoring. If you want to have dense information, a lot of data in an easy, low-cost way that have a good accuracy and good use, you need remote sensing. Accuracy is an issue, and that is acceptable. We know this. There is no perfect protocol. You win some, you lose some. But we need to keep in mind that accuracy has to do a lot with the system that you are focusing on. For example, if I use a remote sensing product that has a cell size of 10 meters and I'm trying to monitor Posidonia oceanica, in Posidonia oceanica, a 1 meter change in the lower depth limit is way significant. And it probably means that I need to take action. With a 10 meter cell size satellite image, I will not be able to detect that change. And again, that is fine, because between us as scientists, we understand this, we know the risks. However, that is not always the case with policymakers. Because for a politician, it's all about having an end product that they can use. And if that product says that there is no change and no significant problem, that's even better. So we need to take care of how we communicate these results. By using the side scan sonar, we think that we have introduced a way to get this accuracy in your final product, especially if you tend to monitor this area again and again, going again to those areas that you have your side scan sonar lines, you get to detect this change. Of course, it is expensive, but you do not have to use it to map this whole region. For example, in this region that I showed you, if I wanted to map it all using a side scan sonar, I probably need something like 4 or 5 weeks. However, we stayed there for 12 days to complete this survey. Finally, no matter what protocol you're using and what methodology, nothing beats the eyes of the researcher, of the person that is there. You not only get your own accuracy, if you like, you feel confident about what you're saying, but also you get the opportunity to identify other issues in the area. For example, in the northern part of that meadow I showed you, we saw a Caulerpa Chilindracea forest, which was way far extensive, and we dare say that it was competing with the Posidonia lower limit. That's all from me. Thank you for your time. Machine learning. Actually, I didn't do it because I'm not a remote sensing person. My colleague who was supposed to be here says not. I'm sorry, though. We can discuss it later on. If we can detect it. You cannot do anything just by remote sensing. There is no way to do that. You will never be sure because, yes, Posidonia has a very specific signature that you can With years, it's something like the matrix. You only see that in a sonograph, but you can never be 100% certain. You need to see it with your eyes, with your camera, with something. Cylindrotraea. No, that wasn't the case. It could have been, but if it was, it wouldn't be Cylindrotraea. It wouldn't be in a high biomass. It could have been, and it was at the lower, at the boundary of the two different ecosystems, and you wouldn't be able to detect it. No, you wouldn't be able. And the reason is that you can identify Posidonia with a sonar because it has air bubbles in the leaves, in the lacunae. So this is what reflects the sound. That's not the case with most seaweeds. You wouldn't see any difference in the image.
To extend the question, you wouldn't be able to this technique for the competition of the two.
No. One is a seagrass, the one is a macrophyte. Yeah. Supposedly, we would have a national mapping program, which is, but we don't. Talking about. Yes. The first data. You need to get the first data. And with Marl, it's way more difficult because we are talking about deeper ecosystems, which means you need stronger instruments, and you cannot use remote sensing. Satellite images don't work there. So you need a proper scanning of the whole of the sea, the whole of the region. Hopefully it will be done. Yes. No, the case study was a part of the sampling stations designated from the ministry for the implementation of the Marine Strategy Framework Directive. And all of this work is being done under that umbrella. Okay. Thank you very much.
And now we're moving to the last presentation of this session, the morning session, is by Dr. Caldatos. Give a talk about machine learning.
Hello, everybody. My name is Dimitris Klaudatos. I am an assistant professor in the University of Thessaly. I was not supposed to make this presentation. This was supposed to be done by students. This is a collective work by students. Basically, it's a case study. We try to see what we can do with just data from published data. Using machine learning or different methodologies. And we've taken it one step further. We have sent a publication now a few days ago. So basically, what we thought that the Mediterranean Sea is the second largest biodiversity hotspot. And we know that from in the Mediterranean Red List, we have about from the 6,000 assessed species, 20% are listed as endangered. And this is basically due to several anthropogenic factors. And our idea, basically, the students' idea, basically, was to identify, to see if we can identify key factors that affect, basically, whether a species is endangered or not. And if we could somehow predict using limited data, if we could somehow predict whether a species in the Mediterranean Sea is endangered or not. So we went about it basically, in the beginning, we thought about doing a pestle analysis, which is political, environmental, social, technological, economical, and legal analysis. We converted to pestle analysis. We changed the political to biological. And we accessed databases and published literature, and we several species, not just fish species, and we basically classed them. We did a binary classification with either non-endangered up to near threatened, the IUCN Red List criteria 0 to 2, and between 3 and 5, we classified them as endangered. And before doing this, I will take you before going this, I'll get back to this. We identified some factors as influential in the beginning, just using basically exploratory data analysis and inferential statistics, and using basically we identified which is like age at maturity is significant. It impacts severely the possibility for a species to be threatened or not, maximum age, asymptotic length, not the depth that the species are located. And we, in the beginning, we try to find some threshold for these identified variables, which is the threshold according to the data we collected for all these species, the age at maturity, max age, asymptotic length. And this was one way we went about it. The other way, basically, we used machine learning, supervised machine learning algorithms. Those, I don't know if you can see them, those 5 algorithms, basically. And we prepared the data. We used as the target variable whether the species is endangered or not. And we did a data split 70-30. We trained the models. And in the end, we evaluated to see whether we can predict, if we can create basically a model that would, with a great certainty and very limited data, identify whether the species is endangered or not. And we ended up with, we used 3 indices in the end. And we basically, these are the model performances. We use the indices, the area under receiver operating curve, the classification accuracy as a precision, and Matthews correlation coefficient. And we basically ended up with 2 models, the random forest and naive Bayes. And this here is the impact of all this data, basically, all this information on the model output. And basically, we managed, as you can see here, the classification accuracy, more than 90% classification accuracy, if we have all this information about the species in the Mediterranean to classify it as an endangered or not. So this was the the results. Basically, our conclusions is that the endangered Mediterranean species tend to mature at an older age. They have a longer lifespan, and the asymptotic length is large. So these are the preliminary drivers we found that affect basically whether a species will be endangered or not in the Mediterranean. And we took this one step further. We collected data for 150 fish species, and we have reached with much less data, only 3 basically information. We can 90% accuracy, we can tell whether a fish species is endangered or not in the Mediterranean. Thank you. Yes, really, yeah. The bycatch data, published literature, whether this was a pilot study, basically. So it was just done by students. This one was quite simple. We started 1 year with just during a course. We had this idea basically to try to find the data, basically data poor science. So without spending any money, collect data and doing some meta-analysis and see how this goes. And the data that the students collected was from published literature, basically, whether this is a species that has been overfished or not. So it was a binary classification, either 0 non-overfished, 1 overfished. There were fish species included, but not only fish species. So this was in the borderline of what basically we wanted to push the limits to see what if this can be done. We didn't know if this would be done. We are preparing something else now with the Bestel analysis as well and several other things. But this was the pilot study to see if this works, and basically using being in the fringe of AI, using machine learning algorithms, you can basically use data that you already have, or they tell you basically nothing, and you can create gold out of nothing. So this is there are students here listening, so this is a new field of exploration, and it's upcoming, the use of AI. Methodology, we have proposed in our publication now the methodology we use. It's a new methodology, basically how you to go about it, because you have to check for correlation between variables. You have to check for collinearity between variables. You have to check for other factors whether you have enough data to do the machine learning analysis. So it's not as simple as I saw it, but this is the first experiment. So it's good enough for a conference, not a publication. This is not good for a publication. So we took it a step further. We have, that's what we have done now. It's more difficult. You get lower classification accuracy with binary. It's more easy for the algorithm to assess. But if you have like a numeric, it's more difficult. And you continuous. Now we've done this with Merluccius, European Hakes age classification. We basically took identified the age from the otoliths, and then we used machine learning to see. We used basically information on the morphometrics of the otoliths, and then we tried to do to see if only by morphometrics of the otolith we can identify the age, and we can with a large about 90% again. So this is a field which has a lot of potential and a lot of uses. So, and as I said, if you have data and you know how to analyze, you can get a lot of output out of. It's poor data science. In Greece, we don't have much money, so we try to find ways to compensate for that. It's a huge advantage, basically, because you can, with the aim is to use as little data as possible to get the best idea of what happens. So if now I have I give you 3 questions about a fish species in the Mediterranean, and you give me correct data, I have trained the model, and I can tell you 9 out of 10 if this species is endangered or not. We did this truthing. I mean, we took some species from the red list that were on the verge, whether out of luck, basically, to see if the model works, and it worked. So basically, it's instead of you can get results much faster using this methodology. You have to supervise it, obviously, but it's a huge advantage. But we cannot rely on AI. I mean, you have to check whether it's right or not, or do the truthing, the same truthing as you did before. Welcome.
Any other questions? Sums up this.
Coffee break.
Thank you all for
So good morning, everybody. I'm Yannis Karakostas, working for a company called Biomar. You can see the logo there. That is specializing in producing aquafeed around the globe. So fish and crustaceans, mostly. You can probably see that it's a global company having production sites all over South America, Europe, Asia, Oceania. And we're dealing with a multitude of species, including species that are very relevant to the Mediterranean market, as European sea bass, gillhead sea bream, greater amberjack, and so on. I'm going to talk a little bit about sustainability today. It is something that we are really keen on advertising and how we deal with things related with the future. Things do not currently look very well, and you can see here that the grade for humanity as far as the environmental assessment is concerned is quite low. It's an F- according to the IPCC. IPCC is the Intergovernmental Panel of Climate Change. And it seems that it will be really hard to live on this planet if we don't do something radical about how things are evolving. So according to the body, it's really hard to deny that human activity is related with climate change. And some not very optimistic calculation states that a number between 3.3 and 3.6 billion people are living in places that in the future years will be highly vulnerable because of the climate change. What can we do about that? What can we do about that as far as the activity related with aquaculture is concerned? You can see here another very interesting point, which is the need of transformation. Until the 80s, you could clearly see that the vast majority of aqua production was related with wild capture fisheries. And by 60s, I would say, early to mid 60s, aquaculture business started to evolve. It was boosted in the mid 80s, and we could see what happened in the Greek aquaculture and the Mediterranean aquaculture in general in the early 90s. And step by step, you can see that the production based on wild capture fisheries had stagnated, and there is a slight declining trend. And quite the opposite is actually valid for aquaculture. By 2020, we calculated that practically 50% of the total aqua production in the world was related with farming of aquatic species. And as you can see, the prediction for the years to come, all the way up to 2050, is that we will still have a steep increase in aquaculture production as opposed to what happens with fisheries. Fisheries are even slightly declining, and this is an added challenge, and I will elaborate a little bit on that. This is a very interesting graph that shows the evolution of salmon feed. We decided to go for salmon because salmon is currently a global product. It may have started from Norway some decades ago, but we do consider salmon right now as a global product. You can buy salmon fillet in ATMs in Southeastern Asia. You put your credit card and you get a nice package of salmon fillet. That means that they have achieved conquering the whole world. This is why we use sometimes salmon in this kind of analysis as a reference point. There is a color coding, as you can see, as far as raw materials are related, and you can see to the right, the legend. And starting from bottom, going up, we have fish meal. I'm going to use my pointer to make things a little bit easier for everybody. We have fish meal, and as you can see in the graph, fish meal in the 90s was a major part of diets for Atlantic salmon, 50% of it, more or less. And then fish oil. Fish oil was responsible for a very large part of these recipes back then, something like 25 to 30%, and a little bit of plant dry matter, what we call vegetable proteins, and starch sources. That was the rest, the other 20%. And we realized that things are not really ideal, and because both fish meal and fish oils are not available in huge volumes. It's a scarce resource, both of them. We decided that the only way to develop aquaculture was to decrease the amount of fish meal and fish oil in the diets. And you can see that for fish oil and for fish meal, there is a very, very significant declining trend that reached its bottom all the way to 2020. You can see this very nice dotted line here. It's what we call the FFDR. It's called forage fish dependency ratio. In reality, it describes how much we rely on marine sources as raw materials. And obviously, if we are decreasing the inclusion of fish oil and fish meal in diets for Atlantic salmon, we have a very significant decreasing trend for the FFDR all the way down to the levels we are today, and what we are expecting that the next few years we will go even lower than that. So we are excluding very scarce raw materials from our mix. What are we going to use to substitute them? And you can see a very significant effect of plant dry matter, vegetable protein sources are playing in this role, increasing continuously, and then a very large part of the fish oil that we were using until 2000 was replaced by plant oils. Soya oil, rapeseed oil, plant oil, other plant oils are mostly responsible for this change. What happens on top of that? We have, because of the removal of fish meal from the diet that contains a lot of very interesting bioactive components, a lot of micro-ingredients start to find their way into these diets, and that's also an increasing trend. And we started quite shyly with single-cell protein raw materials, and it is expected that the contribution will increase significantly. Once again, we will see a few more details a little bit later. This is how a typical diet for Atlantic salmon was in the 90s and what more or less looks like as for today. So as a company, we have some commitment related with our ambitions for 2030. And these commitments are mostly related with three categories. It's climate action. We are committing to reduce the total feed greenhouse gases footprint by 1/3 by 2030, which may sound small, but in reality, it's a very significant decrease. We have committed to include at least 50% of circular and restorative raw materials in their diets by 2030. Once again, I will explain with details. And then 100,000 new people to be directly engaged in capacity building initiatives up to 2030. Let's start with the first one, climate action, greenhouse gases, a very significant part of any sustainability assessment. Feed in a modern society, and as far as aquaculture is related with more or less 80% of the total emissions, and I'm mostly talking about carbon dioxide, but also other gases, from farmed fish. We are trying to reduce the global emissions, and this is related with emissions of scopes 1, 2, and 3. A very quick analysis, and I will come back to my previous slide. Scope 1 are the direct emissions that occur from sources owned and controlled by a company. Company-owned vehicles and facilities. This is what we directly interfere with. Scope 2, it accounts for emissions from energy generation that is purchased or otherwise brought into the company's organization boundaries. So it's mostly the energy that we use in order to produce our aqua feed. And scope 3, it is something that somebody could claim that it's not really of your responsibility. It includes all the indirect emissions that are not included in scope 2 that occur in the reporting company's value chain, including upstream and downstream emissions. And I will go back to my previous slide and I will explain to you that this scope 3 emissions for us are related with the raw materials that we are using in formulating and producing aquafeed. Let's go and see how this can affect our results. These are the actual results we obtained when we did the analysis. It's a relatively old analysis from 2020, but it needs time to be performed. So our global greenhouse gases emissions by then, as you can see, the fragmentation of it, 97.6% are related with Scope 3 emissions, our raw materials that we are using, and only 2.4% are for Scope 1 and Scope 2. So we can improve things significantly in our production facilities, but we will only affect these tiny figures. And the idea is to try to implement these principles in the biggest part of the emissions. So that's a target. Key action on top of our list that is related with raw materials. We work really close with the suppliers to reduce them. We do identify new suppliers, and we drive innovation to develop novel, low-impact raw materials. Once again, I'm going to discuss about that a little bit later. And it seems that, according to the data we have until now, we are the first global aquafeed supplier to commit to the 1.5-degree pathway. So we are contributing for a global temperature increase below 1.5 degrees, which is already a huge task. It's not only us. You can clearly see that this is what is guiding the aquaculture industry in total. And starting from Atlantic salmon, there are several of our customers, salmon farmers, that are trying to perform similar tasks as ours. So as an example, Cermaq is cutting down 35% of the emissions in a collaboration with its suppliers. And Creek Seafood is setting up very similar initiatives by using, let's say, alternative energy sources. Circular and restorative. This is a little bit more complex, and it may require some definition, and this is exactly what I'm going to claim here. 50% of our raw materials by 2030 should be of circular and restorative origin. This is the circular classification. You can see that the product as raw materials classification that we are using here are differentiating between primary ingredients and secondary ingredients. So in the primary ingredient category, we have main products that are quite expensive and full of nutrients, super nice, but with a very significant impact on the environmental challenges, and the co-products that are slightly cheaper, slightly less nutrient dense, but they have a better profile. And then we go for this third category, byproducts, much cheaper, not as dense, but with a very, very attractive environmental profile. If we see a few results, a few details, a few examples, wild fish, fisheries are catching wild fish, and then the main product is oil. You may know the amazing increase in the price of fish oil during the last couple of years. Fish meal, even if it's a key raw material for aquaculture, is a co-product. And then we have all kinds of trimmings and other types of oils that we obtain by using the byproducts of fisheries after the processing for human consumption. The same goes for soybean. You have the oil as the main product. You have the meal or the soybean lecithin as a co-product. And then you have hulls, germs, and gum, and other byproducts that could be used by increasing the environmental profile of the diet. So what does restorative mean? Restorative, as a terminology, raw materials that significantly shift the balance between ecosystem impacts and human production systems toward net positive environmental outcomes. How do we define them? The raw materials that are qualified as restorative, and we have a very relevant graph to the right. It happens when they score a 50% and below lower environmental impact than the most used raw material in the respective category. This is a benchmark from 2020, a little bit old. And in this case, the model raw material or the reference raw material is rapeseed oil. You can see rapeseed oil at the bottom of the graph. And then you make a calculation based on how it deals with carbon dioxide production. And you can see there are a few oils that could be compared with rapeseed oil, canola oil, which is actually another variant of it, or soy oil from USA, or even soy oil from Europe. Soy oil from Brazil is quite unrealistic to be used in order to decrease the environmental footprint. But there is one ingredient in this list, and it's called Alga Prime LS. It's a microalgae-derived oil containing DHA, which is one of the essential polyunsaturated fatty acids that we are using in fish nutrition. This one is scoring 50% lower than the benchmark product. So it qualifies as a restorative raw material. There are others, but this is the oil category, and this is the one that we are looking at now. And enabling people, it's going to be very relevant as far as marine origin raw materials are concerned, and it's about fisheries. So we have improvement projects, FIP for fisheries improvement projects, and this is a very nice example from Mauritania with small pelagic fish, coordinated by key traceability. In reality, what that means, if we are planning to extract fish from the environment, we should do that in a way that it won't nullify the ability of the local population to live on fishing. So we need to collaborate with them instead of being in the other side, trying to find the perfect way, the perfect balance between extracting as much as possible out of the sea in a sustainable manner, and at the same time support, maintain the local communities in their task for survival. This is a very, very big concern of ours. An interesting point here is that sustainability, a few years ago, it was something of, a strange word. People were not really into sustainability. What exactly did that mean? It was one of these marketing gimmicks related with the aquaculture business and so on. What we have seen in recent years, and you can see here a very nice analysis based on European consumers, is that the sustainability is increasing in terms of awareness as far as consumers all over Europe are concerned. And you can see here that sustainably sourced and environmental friendly seafood has a rate which is slightly above price. I can only expect that this may be reversed in the last couple of years after the Ukrainian invasion, but still, the point that we have to take from this graph is that sustainability is continuously increasing in terms of awareness related to consumers in Europe, but we feel all over the world. So, what does consumer want to know about seafood? We did the survey, and 88% of the consumers want better information so they can be confident that they're not buying unsustainable fish and seafood products, which is a massive increase compared to what was the trend in the market 10 years ago. 70% of them, they would like to hear more from companies about the sustainability in the fish and seafood products. And 63% of them, they want to know that the fish that are buying in supermarkets can be traced back to a known and trusted source. And this is really amazing. Some companies can provide that. We're going to see an example later on. The sustainability approach, the design and solutions, and now I'm going to go back to the corporate activity. We do have a concept called BioSustain, which is very much related with sustainability on a corporate level, and we are trying to improve that in any possible way. So, as you can see, there are some layers of must-have in the case of sustainability management, and then some layers that are related with differentiation in sustainability management. And we do think that we are very close to this top part of the pyramid to provide all kinds of information that will assist our customers and the consumers to realize what is the exact activity related to sustainability. On top of that, we do have a concept which is called Blue Impact, and this is a product or service-related sustainability offer. And we are in a position to provide enough documentation to our customers to map and then to record and improve, finally improve, because this is a long trip, their sustainability effect from providing fish to consumers. This is a software tool that we are using, BioSustain. life cycle assessment tool. It's in its 7th generation right now. And we use it for various purposes. It's to make sustainability operational and not just keep sustainability as a generic term, to minimize human errors in the calculation of the sustainability index, to simplify the life cycle assessment calculations, to provide customer reporting. At the end of this tool, we can generate a report that can be handed to our customers in order for them to use it for their customers and provide enough information about how they are committed to be sustainable. And of course, they do provide insights into options for improvement, because at the end of the day, even if a profile is really good right now, the task is to always improve that. And we are using some technology about it. It's compliant with all standards. It meets the PEF requirements, which are necessary now if you want to make sustainability claims. And PEF is the product environmental footprint, which is a new European Union standard towards reliable environmental measurements. And its main purpose is to eradicate what we call greenwashing. You may have heard the term. It's about claiming a sustainable future without having the documentation to prove it. And we have a lot of people in this company that are using this specific tool. It's used in 8 different countries when we have production facilities. There are 19 different employees that are trained to use the tool, and we have conducted 331 studies for our customers since August 2021, when we started to provide this service. You can see it's one of the parameters. This is the global warming impact, and this is expressed, as you can see here, in kilograms of carbon dioxide equivalent. What are we doing here? In this case, as you can probably see, we have 2 diets for European sea bass, 2 diets for Gillhead sea bream. And we are comparing these 2 diets between them and the global warming impact index, depending on if they do include land animal byproducts or not. So you can clearly see, and the land animal byproduct color coding is this pale orange. You can see that it is visible in this code and in this code, but it's absent from the other 2. By incorporating land animal ingredients, which are extremely cost effective and very much available in Europe, we can significantly decrease by 7% in the case of sea bass, or by 20% in the case of sea bream, the global warming impact. And this is a very interesting detail to keep in mind. This is how the tool looks, and you can probably see, now I can see too, that we have several different categories of analysis, starting from climate change and going all the way down to water use. So 12 different categories for the time being, and we are increasing. We had 5 in the previous version, and then we can compare between different diets for the same species, different species, and we can see their full effect as far as the production of kilograms of carbon dioxide equivalent per kilogram of feed used. So a very realistic way to assess the environmental friendliness of the feed that we are producing and our customers are using. And it's not about raw materials on a very generic way. You can clearly see here in the graph that all these columns are related with the very same raw material. This is soya protein concentrate. Where the soya is produced and how it's produced has a major impact in the sustainability index of the product. So you can see under the same category, a generic and certified soya protein concentrate from Brazil scores very highly, which is a very negative point, and we can go as low as 1/8 of this value as far as a very specific soya protein concentrate product from a very specific supplier is concerned. So how we can improve things, it's not just about the choice of the raw material in a generic term, it's about the choice of the raw material concerning the supplier and the production methods that they are using. Blue Impact, I mentioned Blue Impact as a service and as a product before, and I think that we could use this very nice, very interesting example. Lots of talking, but I will try to summarize in an easy way. We do focus on the Blue Impact concept in 3 major categories of sustainability indexes. The carbon footprint that we have already seen earlier on, the circular and restorative use of raw materials that we have mentioned before, and something that we briefly mentioned, which is what we call the FFDR. It's the forage fish dependency ratio. It's a measure of the forage fish used to produce farm fish. In short words, how much fish we use to produce 1 kilo of farm fish. We have created these impact parameters to help provide an understanding on the most critical areas. We had to choose some areas as the most critical. These are the 3 that we ended up with. And our scientists use the most accurate data and robust scientific methodology to provide some figures related with these raw materials. And the plan for all these categories is to move to this blue area. So below 1 kilo of carbon dioxide equivalent per kilo of feed produced, above 75% of use of circular and restorative raw materials, and below 0.5 in the FFDR. So using less than half a kilo of wild caught fish to produce 1 kilo of farm fish. These are the targets for a product or service to be included in the blue impact logo. So we are trying to transition to net zero values. It's not easy, but we have seen earlier that aqua feeds count for 80% of the total carbon footprint of farming. So if somebody has to do something about fish farmers, this is us, carbon footprint. Circular economy thinking, once again, principles and practices that are very, very much the soul of sustainability, and then how to manage scarce resources as fish in the oceans. Good stewardship of the oceans and the aquatic environment is of high importance because we would like to perform this activity, feed producing and aquaculture, for a long, long time. Novel raw materials, we mentioned that also earlier, and we can take a sneak peek into what's next in the future of aquafeed. More byproducts and trimmings. Remember, using raw materials that are byproducts from the processing of fish, mostly for human consumption. More ingredients from single cell technologies. More ingredients from lower trophic levels. We will check on microalgae. And the bottom line, the majority of the diet will still be vegetable-based. Algal oils. It was a huge hype until the day that it became a reality. So what we are actually doing is we are bypassing what is the standard method of producing fish oil, which is microalgae being consumed by zooplankton, being consumed by fish that are fished by the vessels and are producing the fish oil that we are using in everyday formulation for aquatic species. Now we are bypassing all this and we can create some polyunsaturated fatty acids by using a novel method. You can clearly see here how it works. We work in collaboration with the sugar mill. We use the sugar cane to produce sugar, and the sugarcane waste, together with the excess power in the grid, is transferred in the fermentation facility. Microalgae are fermenting into producing polyunsaturated fatty acids. In this very example, it's DHA, docosahexaenoic acid. And why we do that? We're not crazy, but it's more and more difficult to find adequate quantities of fish oil. It's more and more difficult to find enough EPA and DHA, which are the most limiting factors in the planet for aquaculture. It's a real bottleneck. And we can do that by having a very low environmental footprint. They are available on a commercial scale today. They have a cost which is absolutely comparable with fish oil, as opposed to what happened a few years ago. And they are used by our customers in Europe. 4 out of 5 factories that we have in Europe are currently using these algal oils. And it massively reduces the forage fish dependency ratio on oil and the reliance on marine resources. So it's definitely a win-win. Carbon dioxide fermentation to microbial protein. It sounded like science fiction only a few years ago, and now it's in development. We are forming strategic alliances with several stakeholders. In this case, a company called Deep Branch that is producing microbial protein, non-fossil based inputs, no use of land that could be cultivated, nutritional and performance trial being run through 2022. So now we know, and we have the life cycle assessment too, that we can use these raw materials in commercial aquafeed for a number of species. You have probably heard about this story because insect protein is around for a long, long time. And it was greeted in the initial phase as a hype, but things are rapidly changing. So in this case, the example of black soldier fly larvae, we are scaling up and we can use this as a fish meal or a vegetable protein replacer and reducing the forest fish dependency ratio and the carbon footprint. There are a few issues, and the most important one is the fact that current legislation in Europe prohibits certain categories to be used as a substrate. So as you can probably see here, we can use vegetable substrates and formal feedstuff, but no waste. And that would give an amazing boost if ever would be implemented. And you may remember that I mentioned a little bit earlier that consumers would like to have the full traceability of the product that they're buying. This is a very nice tool that one of our customers in Norway was able to develop together with information that we provide through the BioSustain project. So for our customer, Kvarøy Arctic, there is a very nice, very easy to download application for mobile phones that you can target the QR code that is on the package of the fish that you're buying in the supermarket, and you will receive a lot of very interesting information of nutritional qualities, sustainability indexes, feed ingredients, very nice nutritional facts, and how the production of this specific product is affecting the carbon footprint of the industry. I think that this is really a model case, and this is something that we could reach at the end of this trip. I think that we will be able to provide very similar data for any of our customers around the globe, as far as they are totally and fully committed to the sustainability trip that we are also traveling. And this is the end of my presentation. Thank you very, very much. And I'm available for questions. It's not my field of expertise, but I will try to reply as best as possible if you have any questions. We have to define what important means, of course, but I can clearly state that we are about to implement the use of insect meals in our factory in Denmark. So we are very close. And the fact that we are very close is related with there are several suppliers now of insect meals all over Europe. And I'm going to speak for Europe because this is my part of the world that I'm active in. There are several suppliers now that they do provide a diversity of products, high protein insect meals, low protein and high fat insect meals, insect fat. We have several options. I could claim that for 2024, we may not be absolutely cost effective as far as insect meals are concerned. But I think that we are very close, and there are several industries in the market that are upscaling their production. I would expect that in the next 2 or 3 years, we should be in a position that insect meals may be a very standard part of our diets for aquatic species. I would say yes, it will happen. It will happen because of the obvious advantages of the use of insect meals. on top of the nutritional qualities of the insect meals that we have seen that are working quite nice for a number of species, including Atlantic salmon, rainbow trout, European sea bass, and gillhead sea bream, is the fact that the conception of the market is a very positive one. Because insect meals, as opposed to other land animal products, are considered a natural source of nutrients for several of these species. So it's much, much easier to incorporate the insect meals in commercial products for aquatic species compared to the struggle we had with land animal byproducts. So it's not going to happen this year, for sure. Maybe this 10,000 ton benchmark won't happen in the next couple of years, but I'm pretty sure that we will eventually reach this point. Thank you.
Ivana, nice company. First of all, thank you very much for your presentation. And it was very nicely application whereby communicate with transparency your sustainability goals. I would like to ask for the tool for the ACA analysis. Was it based in a standard base? Do you know how it started? Or did you modify the standard base according to your needs? And how often do you modify or update this tool according to your needs?
Okay. When we started using the tool, that was a long, long time ago. No, that was in 2008 or 9. We had to start from something that was not relevant maybe for the full industry, because there were not enough information in the market by then in order to have a globally agreed method to calculate this. But we worked in close collaboration with several of our suppliers, and we tried to use, let's say, the most industry relevant way to make the calculation. Now, there is a standard, at least on European level, that we can use and we can compare between ourselves and the competition. But this actually was applied in this 7th and latest version of the tool. I can probably go back to this graph. It's this one. Here. Yes.
So this is the CO2 for the certain type of
feed. Exactly. You can farm the same species under the same conditions with the same expected performance, but because of the different mix of raw materials, you will have a different effect in terms of the sustainability index.
So this is a diagram.
Very little.
Expensive fish. So this is not the case anymore, but it's still the perception of the people. So the one thing is, how can we change this perception with giving to the consumers the real data that are now in there. And the other one is something that I have also questions from consumers, and it's about the insect meal that we said. We know, because it's our work, that the insect is a normal and usual feed for the species. But when they think about insect meal, they do not understand the meaning of meal as an ingredient for the feed, but they are thinking that someone will feed insects or larvae
Obviously, this is a problem that mostly our customers are facing. So we have to provide with enough information and documentation to them to use them. Once again, I will use the salmon industry as a model. It's going to be repeated so many times that I sound like a broken record, that they can ask for us to provide this sustainability calculation. They can get the full report, and they can get a similar software tool like the one I showed earlier. So every consumer that goes to the supermarket and wants to buy something, they will clearly know that we do not use that much wild-caught fish to produce 1 kilogram of fish. This should happen. I mean, trying to hide it from the consumer, it's a little bit silly. In the Mediterranean aquaculture, unfortunately, the degree of communicating this kind of information to the market is quite low. I have to admit that we were operating more like a cryptic organization than a really open one. So I think that we can massively improve if we can provide this information directly to the consumers. So a campaign with the Farmers Association, I think it would be an amazing tool in order to explain exactly what we do and to show that it's not just the fact that we are already at a very good level, but we are constantly trying to improve things. So not a very easy answer, obviously needs a lot of work, but I really feel that it can be done. The insect meal now, once again, a different thing between salmonids and Mediterranean species. In salmonids, it's actually a natural ingredient and everything is very, very nice. In the Mediterranean area, I've read articles in newspapers claiming that they're planning to feed us cockroaches or something like that, which is not one of the species that we are using to produce insect meal, obviously. How can we communicate that? I think that it will take time. I think that generally speaking, there is at least one part of the European market that is very positive, very open. This is France. And on the other hand, French are very negative towards any other land animal byproduct. But for the rest of Europe, I think the idea of being fed indirectly with insects, it's a little bit strange to say the least. So it will take time. Obviously, it will take time. And I think that the more we communicate the possibility to use insect meals in aquafeed, the better it will come to the consumer's conception that it is an absolutely safe and very high quality ingredient that we can use in aquafeed. And once again, it's this differentiation. Nobody is feeding insects to the fish. It's a completely different story. We haven't spoken about that enough. That is always my reply in this kind of questions. I'm sorry about that, and I would like to contribute as much as possible to externalize what we are actually doing in this industry.
Is somebody else that wants to question the speaker?
Depends on the species, depends on the feed type. But we have seen FFDR values as low as 0.3, meaning that you barely need 300 grams of wild-caught fish to produce 1 kilogram of hard fish. And we have seen FFDR values of 3 in specific species when the nutritional knowledge is not as high. So for yellow-tailed kingfish, let's say that's a very good example, we can have quite high FFDR values because we still rely heavily on fish meal and fish oil for their diets. But for species like rainbow trout, we can easily be below 0.5, and we can even reach 0 if we use correctly the raw materials that are available today.
Sustainable, takes more than one fish, the one
This is a benchmark somehow, a figure of 1. If you are above 1, you are a net feed consumer. If you are below 1, you are a net fish producer. 1 is actually slightly below 1 is the target as far as creating a sustainability story and a reality for this industry. In any case, if we're above 1 in average on global level, we cannot be sustainable. will not be able to continue with the expected increase in aquaculture production around the globe. So definitely, this is where we are today, because it's not, aquaculture is not only about carnivorous species as the one that I have mentioned, bass, bream, trout, and salmon, but there is a colossal production of herbivorous species in Asia, mostly, that are not using a lot of fish meal and fish oil, actually close to nothing. So in average, on aquaculture global basis, we are below 1. The plan is to make even these species, which are regarded as higher value species, being able to dip below this threshold of FFDR of 1. Which species, once again, that's a big question. As an average, I think, I can only speak for myself, I mean for my company, but I think that in average we are around 0.7 to 0.8, which is a very decent figure. The plan is to go lower, and we are working towards it, but at the time we are below the figure of 1, which is already an achievement.
About all this very interesting information that you gave us, and to also remind to the audience that some of these issues are not anymore technological or engineering research. To about the perception of the consumer, about the policy. So if we want to have a less foreign species, it's we have to train the consumer to use more species of fish for not only traditional carnivore species. If everybody wants to eat salmon, bass, and bream, these species in their own normal situation are carnivores. Kept with the fish and the ingredients that they are usually eat. But if you increase the rates that are farmed fish with similar needs or with herbivores fish, then you can wild stocks. This is not any more technical issue, but it's not that you start thinking about perception of the habits of consumers that must be undertaken. Thank you all for this session. Again, by Umar.
Thank you for the opportunity, and I will join again whenever needed.
Yes, on YouTube. It's a bit big, your presentation, 1.2 gigabytes.
Yeah, there's a few videos in there that I usually use, but I think in 101, I skip through some of those.
Good try. Oh, perfect.
It's nice to meet you. Just me. So, yeah, I'm gonna sit on this side also, so that if I say anything wrong, it's gonna get the flag. All right. Yeah. Yeah, the Department of Oceanography, I'm doing the underwater acoustic stuff. But not extracting it, I'm more into these acoustics. That's fish also, but not extracting. So great to hear. Yeah, more about you have an audience, see. What do you about the schedule, your schedule? Yeah, I'm also not used to sitting. Okay. That's a lot of students, you see. So, yeah, acoustics are they're showing quite a lot of interesting. We'll see the exams, though. Don't scare it away. Oh, I am scared by nature, so they're used to it. All right. Thanks very much for you being here and exposing. Couple minutes, it's up to you. Yeah, I mean, if there's an interesting talk on in room A. I think they're all eating the cookies and drinking coffee, so get them inside. I think we will start if you're ready. Okay. All right, so let's start. Yeah, fine. We can wait for a few minutes. All right, up to you. Thanks. She's ringing the bell. She might get a few more. Oh, we have a bell. I didn't know. All right. Let's see if it works. Huh? Let's see if it works. It should be a bell, you know. Oh, that's a bell. The dog house. But we use the mouse to pick to the next slide or the first. They use the mouse to pick through the slides or the arrows? It doesn't matter. Arrows. Anche a queste cose con me non si vedono mai se ne hanno. Modo zicchi. Okay, of course. So, hello everybody, and thanks very much for being here. It's all about acoustic telemetry today. This is a sponsored seminar session type of thing, right? If I, yeah, on fish tracking, I guess. My name is Vasilis, and Vasilis Rigonis. I'm working on passive acoustics and fisheries acoustics stuff here at the Department of Oceanography. Master Shortis, unfortunately, will not be here today. So I'm just going to sit on his side and take his nameplate and give you the floor. Thank you very much for being here. And I just wanted to let you know that you are welcome to ask any question during the introduction, during the presentation. So there will be a Q&A, question and answer session at the end. But of course, you can always ask something or ask to elaborate on anything during this talk. So thank you very much, guys. The floor is yours, and thank you all for being here.
Yeah, thanks for having me again. And yes, as Vasilis said, please ask questions in between. I don't mind to stop in between, don't have a few slides or whatever works. I do have a lot of slides. I know I will sit down in a second. But before I do sit down behind this desk, which I'm not used to, I pass along this tag. You can leave your name, your contact details, whatever. If you like, I can send you all of the presentation because I won't be able to get to all of the slides today, unfortunately. But I can send you a copy, PDF copy of it if you want to later on. So you can do that now or do that later. I will have this sheet of paper down by the booth as well, which is just outside the door. And now I'm going to sit down so that the people online can see me as well. So yes, I'm André Steckenreuter. I work for InnovaSea Fish Tracking. I'm the regional manager for Europe for InnovaSea Fish Tracking. I've done that now for the last 5 years. But I use this technology as a marine biologist myself for the last, well, over 12 years now. So, and it's a very fast developing technology, and you will dig into some basics of acoustic telemetry today. Who of you guys have heard about acoustic telemetry before you saw iTouch talk earlier today? Okay, cool. So not very many. So I keep that very basic here. So I give you an overview and we walk through it. And I'm sure there will be lots of questions that we cannot answer through this session here today, but I will be here until Saturday about lunchtime. And you can always find me at the booth during the breaks and so on. We can have a chat. So my role, there's the first injury of the day. Are you okay?
Yeah. I might need a fish tag on my butt. I'm okay. I don't know how, but I'm okay. That was really theatrical. Okay. That's why Thomas will be sitting here. It's the stairs. Guys, it's the stairs. So, yeah, I'm fine. Sorry for that. Sorry it wasn't on camera. It should be. That will be on my CD.
All right. All right. Okay. If you do that again, you have to pay the beers tonight, right? All right. I'm sure you're okay. All right. So yeah, let's dig into that. And if you do have questions, let me know. This is. Oh, there we go. Yeah, we don't have to go through or I go through this agenda just quickly. So I start with an overview of acoustic telemetry. I go into designing a successful study because that's part of my job. I talk to people initially before they start a project or when they've got their first ideas. I develop research designs with people and then find the perfect equipment for the perfect solution and hopefully good results at the end. I talk a little bit into about some specifics in acoustic telemetry, about specific equipment. I take a few examples out of the whole range that we offer. I go a little bit into range testing, which is kind of the part that you would do before you start an actual study. We're going to go into some examples, a little bit into the software that we offer as part of the package, kind of a little bit of on new technology. As I said, it's a very fast evolving technology and so on. So I do also have a few videos in here, but I think due to time, we have to skip through most of those. So as I said, I work for InnovaSea fish tracking, but that doesn't mean that's all of InnovaSea because we do have a big aquaculture sector, open ocean and land-based, and some aquaculture intelligence, which are basically environmental sensors to measure certain environmental variables. But the connection between the aquaculture facilities or the solutions that we offer and fish tracking is actually aquaculture intelligence, because nowadays we use a lot of the environmental sensors and overlay these environmental parameters with the fish detection data we get. So that's pretty exciting. Gives you a little bit more of an insight. So acoustic telemetry has been around for about 40, 50 years. It all started in the 60s, 70s, really, when the first transmitters and receivers were developed. On the left-hand side, you can see one of the first receivers. I believe inside is a car battery. So you can imagine the size of it and how heavy it was. And now with time, over the last few decades, miniaturization has helped a lot to foster this technology. That means smaller receivers, smaller tags, longer lasting battery lives, and so on, and different technologies in general. Just as a little bit of an overview. Are we there yet? Not, of course we are not, because it's ever developing and emerging, and there's always new technologies coming towards acoustic telemetry, different sensors, further miniaturization, and so on. And we always listen out to the research community. So whatever the research community or you as a researcher need, we try to integrate, if it makes sense, into our existing equipment. And you will see some examples of that today as well. So acoustic telemetry is being used worldwide. Obviously, there are some hotspots that you can see on this map here. This is from a paper from 2015, so it's already pretty dated. So, but it gives you an overview. Hotspots, North America, Europe, South Africa, you see a network there, and Australia, New Zealand, and so on. So there's a few hotspots, but they have been used anywhere from the Arctic oceans to the tropical seas, around reef structures, in very warm waters to very cold waters, in river, in any kind of aquatic environment, really. But I get into that in a second as well. So to give you a little bit of an overview about the technology, we divide the technology in three major stems, and that's active tracking, passive tracking, and real-time tracking. Active tracking is basically when you go onto a boat, and this orange box that was on the left-hand side on the previous slide, on this right-hand side, it's still the gray box, the previous version of this VR100, we call it an active tracking receiver. And that's where you attach a hydrophone to it. And you see the researchers have that pole on the side of the boat. That's where at the end of that is a hydrophone. And then you have to actively track animals. So the hydrophone picks up tagged animals, and the signals will come onto your VR100, and you can actively follow the animals around. Before I should probably go further into it, because I always assume that people know a little bit about acoustic telemetry, but what it's based on is that you've got a receiver part that is listening out to signals, and the other component is the transmitter part that sends out signals. The transmitter emits the signal and is, as you see on the left-hand picture here, is on or in an animal. So you have to tag the animal first before you can follow it around, obviously. And then you've got receiving stations or receivers that you can actively follow animals around, or they're stationary in one position and listen out 360 degrees around this receiver to see if an animal comes into the vicinity. You pick up the signal, and then you've got a time stamp of your animal where it has been at which receiver. If you have several of these receivers along a coastline, around a lake, around a reef structure or whatever, you can then connect all of the timestamps, and that's how you follow animals around, and you can visualize the tracks, and you can analyze where your animal has been at what time, and maybe relate that to other environmental parameters to see, do they prefer warmer waters, colder waters, salinity gradients, whatever you're interested in, right? That's the basic behind it. So active tracking is one way. You have to be physically on the water to track the animals and follow the animals around. So it's for short-term applications, really. That was a ping of one of the transmitters that you heard in the background, and here you can hear that as well. But I do turn that off because otherwise you can't hear me anymore. So passive tracking is a second version that you can use, and that's, I guess, our major applications are passive tracking, because that's when you deploy a receiver in the water, as you can see here. This is a very simple mooring. They can be more sophisticated as well, depending on what you have in mind. But they're stationary underwater, so the receiver needs to be underwater in order to listen to the tags. They don't work as well in air because we use certain frequencies that don't travel well in air, but they travel well underwater. And then you've got an animal that is tagged. Once it's coming into the vicinity again, the signal is being picked up and stored with a timestamp on the receiver. Once you retrieve your receiver out of the water after a certain time frame, that might be a few days, that might be a year or longer, then you download all the data, and then you can connect all the dots between all of your receivers. Then you've got a database, and then you can connect the dots and visualize it. So all of our transmitters have globally unique IDs, and that's very important, because if you, as a researcher, you've got, let's stay with a hammerhead shark here. You've got a female hammerhead shark, you tag it with one transmitter, you know exactly that this ID is only exists only once around the world, because that hammerhead might venture off and go to other places where there might be receivers again in the water, and the ID will be picked up. That doesn't mean that the other researcher knows what animal it is, because you necessarily don't have to share that data with anyone. But you know, once you caught that animal, female hammerhead, it's 1.50 m long, female, and you record all your data, your metadata, and then you've got a globally unique ID. Once you pick up that globally unique ID on any of the receivers, you know that's exactly that animal that you tagged, right? A third option is real-time data, and it's basically the same stationary receivers, but now you've got means of relaying these detections and environmental data as well in real time via Wi-Fi
the mobile phone net, and soon to come satellite connections as well for very remote areas. So you get your detections real time onto your desktop, onto your app or whatever, and you can see which animal has been detected on your receiver in real time. That's the three differences: active tracking, passive tracking, real-time tracking. Obviously, these two slides, this one and the next one, show you a few examples of applications that you can think of. Again, it works in any aquatic environment: rivers, lakes, ocean, coastal, offshore, wherever you can put these receivers or where you're going to actively track those. Some examples are spawning behavior, climate change and pollution impacts, distribution, habitat use, sometimes associated with certain structures like reefs, artificial reefs, aquaculture facilities, and so on. In riverine environments, for example, these applications are not bound to one of the environments. Obviously, they can, they go across these aquatic environments, but they can be migration timing, invasive species research is a very important one lately, nursery areas, feeding habitats, and so on, whatever you can think of. MPA effectiveness, how stationary are certain animals or species in a marine protected area? Are they leaving those? How long are they leaving those? Are they coming back at certain times? Whatever you can think of. We've got three different frequencies that we use. 69 kilohertz is mainly used in the marine environment just because it has got the furthest detection range underwater. And I think you had a detection range question earlier at iTouch talk. So the detection range in 69 kilohertz with our newest receiver model is up to 1.5 kilometers. So if you've got a receiver sitting in the water and it's in pristine conditions, so not noisy at all, then you can get up to 1.5 kilometers of detection range around one receiver. So the animal that comes into the vicinity, 1.5 kilometers around that receiver, will be detected. The noisier the environment is, the less detection range you have. Imagine vessel traffic, different weather patterns, currents, tides, they all generate noise. So the more noise there is, because they're competing with the signal underwater and they potentially mask that signal to a certain degree, then the receiver cannot detect the ID anymore or only parts of it and cannot decode it properly. So to a certain degree. So if it's in pristine conditions, 1.5 kilometers. If it's in less pristine conditions, it's still a few hundred meters, 600, 700, 800 meters, depending on your environment. And that's always versatile as well. It will change over time. It's not always static, like besides you've got a lake that is never changing, but even there will be different weather patterns going through and so on. So it's a versatile performance over time. But you can measure that as well. And I get to that later on. 180 kilohertz, 307 kilohertz. I don't dig too much into that because they're for usually riverine settings, especially designed for hydropower installations where there's a lot of noise in the water due to turbine activities and whatnot. But the higher the frequency is, the better it can cope with noisy environments. And that's what they were developed for. How do you design a study? And that's usually what I go through a few steps here. And one of the questions is, what kind of receiver should I use? And we've got different models. You've got a baseline model kind of next to the orange box here, the VR100, which is for active tracking. But the normal one, and you can see that outside as well, it's like a 1 liter bottle kind of size. Then you've got a different one with that little collar there. Can you see my pointer there? Yeah, okay. So this one here with that collar is basically like the basic unit, but integrated with an acoustic release. So with the orange box here on the left and the hydrophone attached to it, which is not on this picture, you can talk to these units and you can tell it to release the bottom part, this lug at the bottom, and it screws out. And once you've got a positive buoyancy here, you've got a float on top of that, it comes to the surface, you can pick it up and you can download the data that way. So it's an easier way of getting back your receiver if you can't dive to it or if you can't retrieve it in any other ways. The one in the middle here that I usually don't carry around with me because it's heavier and bigger, it's a dual frequency receiver. You can see there's two hydrophone tips here at the top, one in 69 kilohertz and one in 180 kilohertz. And that's not the only important thing here with this receiver, the VR4, but you can also, the download the data via this modem with a certain hydrophone. So you can leave this unit down at the bottom of the ocean for up to 8 years. It stays there, and you can only go there with a small boat, chuck in a hydrophone, a certain hydrophone that can suck up your data through the water column straight to your laptop, and you don't have to touch the unit. Obviously, that can save you a lot of money and time that you would otherwise spend on field work. And if you don't need to retrieve the units, then that's one way of saving money for long-term projects. Obviously, if you've got shorter projects, one of the basic units are cheaper, because the dual frequency one where you suck up the data is about 10 times as much in price. But for long-term projects, it's worth considering those, because you can save a lot of money for big ocean vessels, lots of diesel fuel that you would spend otherwise in the open ocean, and so on, and lots of staff that you would have to pay as well. And I always say the easier the mooring, the more streamlined it is, the easier it is to use, the less can go wrong, and so on. So, but whatever fits your application, right? I don't go into that in too much detail here. Battery life, we've got different versions. Some of them last 3 to 6 months, because you can use alkaline batteries or lithium batteries with the better yields you get with lithium batteries, obviously. And as I said, on the right-hand side here, the VR4 can last up to 8 years without rebatterying it. But you can always rebatterying them yourself. You can buy the batteries from us or from other manufacturers, and you can just get them out of water again, screw them open, put a new battery in, and they are good to go again for, so the baseline units here in the middle, they all last like between 13, 14 months at least, and then they need a new battery. A few examples of mooring designs. Again, I always say the easier, the better they work. Like let that be like a metal pole here with a concrete filled tire or whatever you can think of. Some more sophisticated mooring designs that are also attached to kind of a cable or something. They are especially used in front of hydropower stations, and the cable is therefore, so if the unit is dragged around with a current or high flood events, they're not ending up in any turbines, for example. So you need some secure measures. You can see one of the units here, the VR4s, being mounted upside down at an artificial reef, for example, at about 30 meters depth. And to listen out not to the top of the water column, but here the researchers wanted to see which animals are below the structure of the artificial reef, for example. And here I skipped through this video, unfortunately, because we don't have time for it, but we can have a look at it again at the booth if you're really interested in it. It shows you how an acoustic release receiver is working, how it's deployed, and how it's coming back to the surface. Here's a little animation as well. The acoustic release receiver is in the middle of this collar here. Still the same unit, but now you've got a collar around it and integrated floats and integrated with a rope can so that you can retrieve the anchor at the same time. If you have the unit only with positive buoyancy, it comes to the surface and leaves the anchor behind because the little screw at the bottom screws out and the anchor sits at the bottom. To counteract for that and to retrieve the anchor at the same time, you can add this arc or acoustic release canister that integrates the floats and some rope. And once you torque to your unit from the boat to a unit to release the bottom screw, it comes to the surface, but spools out the rope at the same time. So you're still connected to your anchor. Once it comes to the surface, it flips upside down. So there's still communication between your hydrophone that you have on the side of your boat and the receiver hydrophone here. So you know how far it is away from you, for example. You see the orange floats popping up at the surface, so visual cue, and then you retrieve your unit first, then you pick up the anchor with a rope that has spooled out. So you retrieve the whole unit and don't leave anything behind. There's also mobile alternatives. So we were talking about stationary moorings previously, but there's also some mobile alternatives. That can be a sail drone here on the left-hand side, wave gliders, slocum gliders, and so on, where people have attached receivers to, and then they go on certain expeditions with those mobile platforms and go through gyres along the open ocean, where you can probably not deploy a mooring, a static mooring, where it's too deep, where it's too rough, and so on, or where you don't have any permits to deploy anything on the bottom of the ocean. So those are some alternatives. I'm also working with some other researchers just now to attach some of these receivers on a sailing boat, converted into like an expedition boat. So if they go on expeditions and they've got the receiver attached to it, they know what they can pick up along the way, for example. So that's kind of a different version of it, I guess. It's kind of a mobile tracking, but in a different version, I guess. And once we decided what kind of receiver you would like to use, or you can talk about transmitters or tags first as well. If you've got a certain species in mind that you would like to tag, then you know how heavy it is, the size of the animal, where it lives, and so on. And then we can determine the transmitter part as well, which size fits best, which frequency do you want to use, and so on. And then we've got tags that last from about 6 months with our smallest tag, the V3, which is 0.3 of a gram, almost as small as a pit tag. And then the largest ones, the V16 here, all of the numbers beside the V are the diameter of the transmitter. So 3 is 3 millimeter, 16 is 60 millimeter in diameter, and the largest ones last up to 10 years. And you can imagine they're especially for long-lived animals, sharks, rays, turtles, sturgeon, whatever you can think of, right? And they're usually used for long-term applications where you want to see movement for many years to come. The smaller ones you use for either small species, juvenile life stages, where you wouldn't otherwise be able to fit a transmitter on. And that's our smallest one. And you can tag animals that are as small as 6 to 15 grams. That's the smallest one. As part of the transmitters, I told you earlier that they always have a globally unique ID. And you can also decide if you want to have some sensor data attached to it. So you have the ID plus some sensor data. And that can be temperature, depth, acceleration, and predation. Temperature is clear. You measure the temperature of the environment the animal is in. Depth, at what position it is when it sends out the signal. Acceleration is important, for example, when you want to detect hunting spouts, feeding behavior, mating behavior, and so on. It's usually used for that, or how fast an animal is moving through a certain habitat, migration-wise. Predation, I will give you that example now because it's one of our newest technologies. So some of you have been out to the booth here where I have some of this on exhibit. So the tags are usually very inert, black kind of tube. They don't leak. They can last for many, many years. But you can also, there's a little white polymer if it's a predation tag. And that little white polymer, and I'm going a little bit ahead of me, but here you can see it. So here you can see that little white polymer on all of these tags. And you tag an original animal with your original tag. You've got one ID. Once it's been predated by a predator, no matter what kind of predator it is, this little white polymer will dissolve in the presence of the stomach acid. Once that has been done, there's a little magnet inside that flips a key inside or something like that, and then it switches ID. So now you've got a different ID. Your original ID might have been 1, 2, 3, 4 for original animal that you tagged, and now it's 1, 2, 3, 5. And now you know when you get a detection of 1, 2, 3, 5, you now know that you are following the predator around that might have a totally different behavior and totally different movement patterns than your original animal. They were originally developed for salmon smolt, so juvenile salmon work, because there were researchers finding erratic behavior of the salmon smolt. They would think that the salmon smolt would go straight downriver into the ocean, but then all of a sudden, maybe near a bridge, maybe somewhere else, it would go upriver again, and there were erratic movement around, a bridge or some other structure or something. And then they said, well, we assume there was some predation happening because we believe that we are now following the predator around, but we could not prove it. So then we developed this new sensor technology, and that's what I said before. We always have exchange with researchers. What do they need? What's important for them to understand? So if people say, well, we've got salmon smolt that behave differently than they are supposed to, is that behavior, like, is that normal behavior of the salmon's mode, or is it not? And then, because predation was one of the factors that could influence that behavior, we designed this new predation sensing technology. It's very important to see what kind of transmitter fits your animal. So usually people tell me, oh, my animal is like 30 or 40 centimeters long. The important bit here is really the weight. How heavy is your animal? And because we are looking at the weight, animal weight to tag weight ratio, and that usually should not exceed 2 to 5%. So with the smallest tag that weighs 0.3 of a gram, you can tag animals that are as small as 6 to 15 grams, right? And then, you know, to the top, like whale shark is another extreme example that you can tag there. It doesn't matter which kind of transmitter you put on, right? But yeah, so you want to have a look at the weight of your animal. It's obviously different sometimes if you work with crustaceans. I always talk about fish, but we will see in a second that you can tag a lot of aquatic animals, right? Let that be crustaceans, invertebrates, or whatever, depending on their size and where you can fit a tag. But there's always ways of finding a way to tag most animals. Otherwise, unless they're very small, you know, below, let's say 6 grams. Usually they're tagged, let's go to the next slide. So usually here there are fish examples. So usually you want to have transmitters that are internal, you want to tag internally. So you have a little incision on the body ventrally, and you put it into the body cavity and suture it back together, 1-2 stitches. And then the tag retention is much better because they're less prone to fall off, to have biofouling on them or whatever over time. Imagine you've got a 10-year tag, how much biofouling you might get over the years. But internal tagging is preferred, but sometimes there's only ways of externally tagging, or sometimes it's preferred for certain species because they can't handle internal tagging. But there's always different ways that you can use. One that stands out here to the bottom left, where researchers converted an original cattle tag that goes into the ear of sheep or cow or whatever, and they integrated an application where they could fit a transmitter into that original cattle tag and then put it through the dorsal fin of sharks, for example. But there's always like different ways around it of how to tag animals. Talking about different species. So again, if you do have questions in between, don't be shy, raise your hand, shout out. Other tagging examples are sea turtle hatchlings, adult sea turtles, tortoises as well, freshwater turtles, lobsters, seals as well. I think you asked about marine mammals, if they can be tagged as well. Yes and no. 69 kilohertz as a frequency in the marine environment is hearable by a lot of cetaceans, for example. So usually people shy away from using 69 kilohertz on marine mammals like dolphins, for example. But seals, they have their hearing range in 69 kilohertz as well. But I don't think it changes any behavior, and there's no papers about it. And the people who are working on it don't seem to see an effect if you tag seals, at least. For dolphins, I don't know, because I don't think there have been studies on it. But seals are tagged on a few occasions. If you are worried about 69 kilohertz being a frequency that is that the seals can hear, you can always switch to 180 kilohertz, which is again then outside of the hearing range of seals, for example. That's just on the side. Crustaceans as well, you can tag. You have to just be aware of their molding processes and intervals to tag just post-molding to get the longest transmitter live on these animals. And then the animal to tag weight ratio doesn't necessarily mean to stay between the 2 to 5%. Sometimes you can go beyond that as well, because they are, they don't depend on hunting spouts where they have to be very agile, like a bonito or tuna or shark, for example, or whatever else, or herring. And crustaceans move slower and so on. So you can use different ways of tracking them. I see I'm always half through this seminar, so I'm trying to do my best to go through most to a lot of slides here in the next half an hour. But again, don't worry if we don't go through all of the slides. I can send you the slides later on, whatever we can skip through, and we can have a chat at the booth and so on later on as well. I mentioned environmental sensors before as well as part of our aquaculture intelligence component under Innovasi, and now they're feeding into the fish tracking realm as well. So you can, the nice thing with these aqua measures, they can be used as a standalone unit. Once you deploy them and you retrieve them again and you want to get your data off, you can use a mobile app on your phone to download the data. But the other way is to push the data to your acoustic receivers that are in listening range around these acoustic, around the aqua measures, the environmental sensors, and they can push the data straight to your receivers. And then you've got your animal detection data combined in one file with your environmental sensor data. Very easy to analyze, and you've got all in one. So, and aqua measures always have temperature as one environmental variable and one other sensor, like DO, dissolved oxygen, salinity, turbidity, chlorophyll, and whatever Turner electronics that we collaborate on those, whatever sensor they develop, we can integrate in aqua measures. And then it's pushed either to the app on the left-hand side here, or on the right, or on the right-hand side, I should say. And on the left-hand side, it can be stored on your receiver, just as a, and I've got an example sitting outside as well. Once you open up your app and you downloaded your data, you can see the first visuals of your data. So you see an oxygen curve here and a temperature curve, and you can push that to the cloud or store it somewhere else, whatever you're interested in. And then you can combine it with your detection data, and then you can answer not only the question, where has an animal been when, but also potentially why. Does that depend on temperature, salinity profiles, and whatever other sensors you might be interested in, or environmental variables. An example of transponding receivers I want to give you, because that's kind of our newest technology in terms of receivers, that they not only get the detection data of your animals, but they do collect a lot of other very important diagnostic features, for example. That can be noise around the receiver. So you can download the noise profile throughout the time it has been deployed. And that helps into interpreting your data later on. If you, for example, didn't get any detections at night, that doesn't mean necessarily there weren't any animals around. It could have been too noisy. And then you can have a look at the noise profile. Was it very noisy at night? Very typical around tropical reefs, because there's a lot of biological activity around tropical reefs at night, a lot of feeding activity and so on. So the noise levels are usually rising at night, and maybe the detection range decreases with that. And through the noise file, you can have a look at if there's a pattern to it, or at certain times when there was a construction site going on, aquaculture activities like feeding activities in certain intervals, you can detect those and say, okay, maybe I did have animals around, but I couldn't detect them at the time because they weren't in detection range of my receiver. Tilt is another one. If you've got not a static mooring, but it can sway in the currents with the tides and so on. If you've got an anchor, a rope, and a float, and the receiver sits somewhere on the rope, it can sway in the current and so on. And you can have a look at how that affected your mooring over the deployment period. And if you look at the tilt, it was 90 degrees, it was lying flat on the ground, you didn't get the optimal detection range around your receiver, right? Temperature is another one that you can have a look at the profiles, and so on. So that's the advantage of transponding receivers. They also have internal transmitters, and that has got a few different reasons. So the transmitters that you put on your animals or into your animals are the same, or it's the largest version of those. The V16 sits in these receivers as an internal transmitter. So the receiver can also emit pings, and that has got different reasons. You can use it as a sync tag. That means if you've got, you can see a triangle of receivers. And if you're interested in not only presence or absence of your animal around the receiver, so if you've got a singular receiver, you will detect presence or absence. Your animal has been there or your animal has not been there. So you detected a ping or you didn't. If that's not enough for your application to answer your research question, you don't want to know if it's 360 degrees around one particular receiver or mooring, but you want to have an exact position of your animal, you need at least 3 receivers overlapping with their detection ranges, because then the signal of your transmitter will be detected on each of the receivers at a slightly different time. And the time of arrival difference at each of these receivers, from that you can triangulate the exact position within that triangle, or a little bit further beyond that, where the exact position of your fish was. So you need at least 3 receivers. Most people use 4 receivers, so they've got different triangles that they can choose from. If one receiver didn't hear it, the likelihood of the other 3 receivers listening to it and detecting it is higher. And then you can position the animals to a better degree. In 69 kilohertz, which we mainly use in the marine environment, I always combine it or compare to a GPS signal, a few meters resolution. The higher the frequency is, 180, 307 kilohertz, you can go, the best solutions that we had, or the best calculations, were 10 to 15 centimeters accuracy of a position of your fish within the water column. 2D, 3D, if you want to take the depth into account. And the internal transmitter, that's where I was, helps you to synchronize the internal clocks of your receivers, because you need that difference of time of arrival of your signal. And then all of the 3 clocks of your three receivers need to be exactly the same. Otherwise, you know, the positions will be skewed and being less accurate. That's what it's used for example. But you can also use it as a range test tag, and I get to that in a second. Sometimes you can also use it as the sync tag. If you've got one receiver sitting somewhere, you still want to leave the sync tag on that emits a signal every, let's say, 5 to 10 minutes. In case you're losing your mooring is shifting due to fishing equipment being entangled, being a flood current happening, a flood event happening, or whatever else is happening. Sometimes your receiver, your mooring might have been shifted. And this guy, Sam Schrey in Sweden, he would take one of the VR100s, the mobile tracking receiver, and a hydrophone, snorkels down his river where he is doing his research. Once he gets a ping from his internal transmitter of his receiver, he will get a ping and he knows it's in the vicinity, then he can look for it. And here you can see it lying on the tree here across that body of water. And here he found his lost receiver back again. And I've done it myself, so it works very well. Before in the older models, we didn't have these internal transmitters. Once you lose a unit, yes, of course you can look for it. And I used like underwater metal detectors or whatever that you can see to find it again under sand or something, but it's much easier if you get a signal from it. Don't get into that too much. A little bit into range testing, just because it's so important if you start a new study. Before you start a new study in a new environment where you haven't done any acoustic telemetry before, you don't know the noise profile of your environment, we always tell people, do some testing beforehand. And one of the things that I do with researchers as well, if you've never been exposed to acoustic telemetry before and you don't know how it works, I do a lot of workshops and trainings on site with people. And we go into the field, we deploy some units, some receivers. We've got some range test tags that we take with us, and then we deploy a receiver. And then we see how far can the receiver detect these range test tags in your particular environment. Let it be a fast-flowing river, let it be a coastal remote area where there hasn't been any acoustics being done before. We can basically test your environment first, and then you know which kind of equipment works best and what you can expect from your results later on. Usually people incorporate that into their budget, and they say, well, we want to do range testing anyway, because a lot of scientific papers, if you want to publish your data later on, they want to have this baseline data of you first. They want to see, have you tested your environment first? What's the 50% detection rate, for example, in your environment? Where do you get every second signal? And that's your baseline data, right? And we can do static range testing, we can do drift range testing, but there's a lot of different parameters that we collect along the way. well, here's a setup of, let's say, a few receivers. You put those in the water. You've got a test tag on the right-hand side here. And the further you are away from this range test tag, the less percentage of signals are reaching the furthest receiver, right? So you will get a curve that looks like this at 0.0. You will get close to 100% detection or 100% detection rate. The further you go away from the range test tag or you tag animal later on, the furthest it is away from your receiver, the less likely it is that you get all of the detections. And usually then we have a look at the 50% mark where you get every second detection. That's a little bit of a background. That might be a little bit too much for this audience, but it is very important. For example, if you want to have a receiver array design that is a gate or curtain, and all of these dots here, no matter if they are red or the purple circles here, if you have a gate across a body of water, let that be a river or an entrance of an estuary, for example, if you put receivers across and you expect them to have a certain detection range, you can either have a leaky gate so they don't overlap with their detection range, so animals can pass through these receivers being undetected. That's what you don't want to have. And that's why it is very important to do range testing to see, especially for these applications, that they overlap with their detection range so that you don't have a leaky gate, but that you get all of the detections of animals passing through this curtain or gate of receivers. A little bit more of a background on that. Fine scaling positioning, I explained already before, so we can skip through that. I want to give you some examples. So we talked about research design. How do I understand which kind of receiver works best? What kind of transmitter do I want to use? I did some range testing, so it works in my environment. Now I deploy my receivers in the water for a certain time, let's say for a year. I tag my animals, and now I get some data back, and now I want to analyze it. So first thing, we have visual animals. We get a lot of information out of visuals. So what people like, or we as humans like, are tracks and some visuals to see what's happening with our data. I want to give you some examples on all three frequencies to see what's kind of possible with acoustic telemetry. So here's an example of sea turtle hatchlings, and there's several of those studies being done. One of the first ones were done in Western Australia, at the north coast of Western Australia, on flatback sea turtle hatchlings. And you can see on that picture here towards the bottom, they had the little tag. This was the smallest tag in 69 kilohertz, in 180 kilohertz at the time. And they had it horizontally below the vertical part of the sea turtle hatchling to start with in a pilot study, but they realized they don't get perfect signals being sent out from the sea turtle hatchling. And then they put it vertically below the sea turtle hatchling here. Obviously, they cannot leave their nest and crawl along the beaches with that being tagged. So they're released in very shallow waters just off the nesting beach where they are born. And then you can see in this study, there have been some artificial light sources like these orange diamonds here and there. So in ambient nights, when there was no artificial light, you can see the animals are moving straight off the nesting beaches. There's no artificial light in the background that is disturbing them. If you introduce an artificial light source, you can see how these tracks are being skewed and that the sea turtle hatchlings are moving towards that artificial light source. So these ones are the little asterisks here, or the stars are the release side. And then all of the little white empty circles here are one receiver. So they've got an array of receivers, a square. The receiver distance of each of these circles is 50 meters in this study, because they wanted to have overlapping detection ranges of many of those receivers, so they can position the sea turtle position very accurately and see the tracks where they're going. One example. And here there's an animation of exactly the same thing. You can see each of these different colored tracks is one sea turtle hatchling, and you can see how they're moving through these receivers. So all of these numbers here are receivers, and you can see the grid of receivers of that nesting beach here. This is a study, again, on 180 kilohertz on lobster. And here you can see a coastal array with lots of the receivers overlapping with their detection ranges. And then you've got some singular receivers to see if there's any lobster movement out of this core area into other areas that are not part of this core area that people were interested in. This might have been an MPA or something where the lobsters are hiding mostly and are using the habitat mostly. But then there, at certain times, you can see them venturing off to different parts outside of this core area. Just checking the time. I go through a few more examples here. This is a lake study, and here you can see a 3D kernel density analysis, a lake in Canada. This is data of 1 year. Each of these little dots, different colors again, different individual fish. This is brown trout in a freshwater lake. And you can see where the animals are positioned over the course of the year. And I can tell you from the analysis from that paper, there is one deep spot here, which is the wintering ground of the brown trout, where they're hiding over winter. And during warmer months, again, they're venturing out and using most of this lake and go to shallower areas to where there's probably preferred feeding habitats. As an example. Here's a hydropower example on 307 kilohertz, where you can see that, look, this is the hydropower. These are all the turbines. And then there's a fish passage device here, and this is the same thing. And you can see the entrance of the fish passage here, and you can see salmon smolt coming from upriver. They want to move downriver, and they were expected to find the fish litter, and most of these did and didn't go through the turbines and use the that way. As another example for fine scale positioning. This is an eel study, again on 307 kilohertz, and you can't see that on this visualization, but the fish ladder in this particular installation is positioned around here. And this was a pilot study. I think they tagged 8 or 10 animals, eels, European eels, and they were all going through the turbines and being shredded, as another example. And you can here see very nicely that you can not only do that 2D animation, but you can use the third dimension as well, depth, to have 3D analysis and visualizations as well. Here's an aquaculture example. I think there's 2 in these slides. So there's some people interested in fine scale positioning of animals within aquaculture facilities, let that be sea pens or concrete tanks or whatever. And you can see inter-animal movements, inter-animal relationships. You can optimize feeding strategies. Are most of the animals at the surface for most of the time? Are they using more the bottom part of the sea pens? And there's more and more aquaculture acoustic telemetry studies popping up. And I'm talking more and more to people utilizing acoustic telemetry in confined areas as well. We're talking to some people who want to do acoustic telemetry in big aquariums, talking sea life aquariums, to see how animals are moving in aquariums. Are they disturbed by visitors, for example, and the noise that visitors emit in front of those big aquariums, and so on. So yeah, lots of different applications. These are just a few examples. Some 69 kilohertz examples. I lived and worked for a long time in Australia as part of the acoustic telemetry network in Australia. It's all based under IMOS, the Integrated Marine Observing System in Australia, and they've got a long-term database for fish tracking or for acoustic telemetry tracking. And this is data, combined data from 2009 to 2016, so 7 years of data of 7 different shark species and how they're moving along coastlines. And you can see a very nice animation here, zooming into some parts of the Great Barrier Reef here. So network means, so you as a researcher, if you put receivers in the water, your receivers, you tag some animals, you've got your IDs that you know on which animals they went onto or into, and you get your data later on from your receivers. If you've got animals and species that are moving beyond your receivers, you usually don't get any data, right? And that's when you want to collaborate with other researchers in the area or where you expect your animals to move to. And this is exactly what happened in Australia. and what Ichthyology talked about in the Aegean Sea and the Black Sea, that people are coming together to utilize different people's research receiver infrastructure, so that you get notified when your animal has not only been detected on your own receiver, but also on someone else's receivers. And then you can have a database like European Tracking Network database, the Australian equivalent, the IMOS database, and that's where all the data comes together. And if you put your data in there, if you are working on Port Jackson sharks, for example, very good example, they were working in a marine protected area of Jervis Bay, Marine National Park, because the Port Jackson sharks were there for their pups to get their youngs in the protected bay. And they were there for several months a year, 6 months, 5-6 months a year. They were stationary there, rearing the pups. They were born there, and then the adults would leave the enclosed bay and would venture off. Nobody knew where they were going for the second part of that year. Once the researchers put their animal data into that database, they realized, oh my God, they're actually, because they are sedentary animals, benthic shark species, usually not known to move long distances, they were moving from around that part here all the way to Tasmania and the Tasmanian Strait here to feed on benthic crustaceans mainly and so on. And then they feed for half a year, then they come back over hundreds of kilometers back to where they rear their pups and where they give birth to their offspring. So they would, and really every researcher I talked to over the last 12 years or so, whenever they start putting data into some kind of these databases and they exchange knowledge of their transmitters and so on, they will always get some surprises where their animals are popping up. So that's kind of one of the most interesting things that I come across when there's something new popping up, when you not only count, your animal is here, it's moving around, it's using the seagrass meadows for a certain time, all good. It's all necessary and it's all good research. But for me, the really interesting part is where are your animals going when they're not within the detection range of your own receivers. I think that's just, my kind of secret hobby. Real-time data, one example for real-time data is that you can get real-time data of tagged animals, and again, an Australian example, because it hasn't been done in many areas yet. There's another Californian or West Coast example, or in North America, where they do similar things with different species. But here, the National State of Australia and the fisheries there, they are tagging 3 different shark species, especially white, bull, and tiger sharks, because they're mainly attributed to shark attacks at swimming beaches, surfing beaches around Australia, and they wanted to tag as many of these 3 species as possible. And this has been going on for last few years, I want to say 3 or 4 years, and by now this number is outdated already. It's I think beyond 2,000 sharks now of these 3 species along the east coast and the west coast of Australia. And then once an animal comes within detection range of these live real-time data receivers, then you can freely download this app. It's called SharkSmart in Australia, and then beachgoers, lifesavers, and whatever at the beaches can then get notified of the tagged animals being around one of these real-time receivers along, the coastline. And then you can decide if you want to go in the water or not. Obviously, this is a political decision as well. Before that, most of the surfing beaches in very populated areas around Sydney, for example, Newcastle, and so on, they were netted off underwater. There were nets that were trying to net off beaches that the sharks would not approach the beaches, but they're basically like tennis court nets. They were open on either side, up and down. So they would entangle lots of aquatic species, sea turtles, fish as well, sharks, marine mammals, and so on. And I think this real-time acoustic telemetry application, even though it's far from being ideal because you can't take all of the animals of these 3 species, for example, in a certain environment, or at least not over the course of a few years,
but I think it's at least a better solution than putting nets, underwater and catching whatever is getting entangled there. So that was a little bit of an overview of some of the application examples. I don't think we will go through the software suite here in terms of time because it's almost 2 o'clock. But if you do have questions, please leave your contact details on the sheet of paper that I pass around here. Come to the booth, have a chat with me. You can take one of my contact details. You can also see my contact details at one of the last slides here. And take a picture of that or come and get one of my cards at the booth. And you can send me an email if you do have ideas, if you want to start a master thesis, PhD thesis, and you are interested in acoustic telemetry. We also have some student offers where you can get parts of this equipment for a reduced price, about 30-something% cheaper than the original price, for example. So that's an incentive for research groups to incorporate students' research as well. Yeah, just come and have a chat. Thank you very much.
Thoughts, ideas, yeah, please. I want to ask what was the name of the project used to really track
which one? Sorry.
You showed us a program that was 3D.
Oh, 3D movement. Yeah, there's different projects using 2 or 3D animations. That was a particular pilot study of the eels in front of the hydropower or the brown trout in the lake. There are different institutes, different universities, fishery agencies, and they either do the data analysis and those visualizations themselves, or we have a consulting team. So everything produced by Novosea is produced in Halifax in Canada, on the east coast of Canada, and then it will be sent out to different institutes around the world. And but yeah, it's all manufactured there. But we have a data analysis team as well that can, if you are not confident on analyzing these kind of data sets, you can either get some help and training from us, or you can just send in all your data. We analyze it for you, obviously for consulting fee, but you can always integrate it into your budget if you like, or whatever. Some people are not confident in analyzing that data, and then I think it's a good way to make that happen for people who have never tagged a fish, for example. We do lots of consulting services as well. We can tag fish, we can deploy receivers, we can analyze data, we can do the whole projects. We do that mainly for hydropower companies because they usually don't have biologists employed, and then we do everything from A to Z, and they get a report at the end, and they know the results. Or we just have researchers along to see where they need help and training, and that's one of my aims as well, to go to different institutes, fishery agencies, NGOs, whatever it is, and to tell them what equipment works best for kind of the solution that they're looking for. Any other questions?
They're looking at me. You should look at me. Yeah. I just wanted to add that I can reassure you guys that you only can cover a small percentage of the applications of this technology, and it goes all the way out to the open ocean, tunas, fish aggregating devices, tuna fishing in the open ocean, all sorts of stuff combined with other sensors and everything. So it is a very widely used technology. And my question now is, from your experience, how much does the tagging itself modify the behavior of the animals? So for example, for a tuna, you grab a tuna, you cut it open, you put it out, you put some stitches, and the tuna is back. And I was part of such an experiment. We were actually looking how tunas were moving around fats, drifting fats in the Indian Ocean. So, and there was always the question next to predation, which is solved now. How much was the behavior, modified? Because
every time you tag a new species, mainly there's some lab studies or some pilot studies being done. If you tag a new species, for example, I've got one university who is interested in tagging archerfish, you know, mangrove fish that shoot out a water spout and to get insects off the mangrove roots and so on. Have never been tagged before. So now they're doing a pilot study first to see how well, so we are staying in between these 2 to 5% animal weight to tag weight ratio. So we choose the right transmitter, but some species behave differently than others. And some species can be tagged easier than others. Herring, for example, don't like to be handled, or shad, they don't like to be handled and they keel over fairly easily. If you've got a bonito or, you know, some sharks or whatever, they're not bothered at all, you know. And usually all of the aquatic organisms have a physiology that they heal very quickly. You know, that a wound is not staying open for very long to minimize infection rates and so on. So tagging has been done with many different species. If it is a new species, you want to make sure that you don't alter the behavior, because you want to detect the normal behavior of your species that you're interested in. And if you don't know, you do it in an aquarium or in a confined area where you can see. You can also use dummy tags without any of the technology inside. Same weight, same dimensions, but are much cheaper, obviously, because there's no tag inside. So we've got some of those dummy tags, and then you put that into an animal or onto an animal, and then you see if it's working. First of all, the weight ratio. And if you are confident that it's working, you might use a live tag that you put in a tank, and then you see how it's handling it over maybe a course of a few weeks, a few days, a few months, whatever you want to do. And you have to be confident as a biologist. First, if I release it in the water, you don't want to drop it to the ocean, bottom of the ocean, because then you don't get any data back. So yeah, but those are kind of the techniques what you do. And then usually there are lots of scientific papers out there that you can look up how a tag might or might not alter certain behavior. Yeah. But usually it's the percentage is that small that usually that's not really a big issue.
Hey, can I have another question, or is there someone from the audience? Yeah. I'm not an expert in falling spectacularly. I was thinking applications in our waters, like shallow waters, coastal waters, combined with hydrophones and whatever. And okay, my question has to do with that graph that you showed with the little turtles and the artificial lights and everything. So my question is this one. The detections of the little turtles, I saw that per color, they were happening in batches. So they were either not being detected when they were up to breed, or I don't know. So my question is, apply this technology in shallow waters, MPAs, coastal MPAs that we do have for acoustic diversity, fish diversity, combined with other hydrophone sensors. Am I more likely to lose fish that are shallower or deeper in shallow waters? Because if I'm right, then the turtles were not being detected continuously. So you either lost them when they were up to breed, or I don't know if you saw that. In that graph, no line was continuous. They were present in batches. Then plants.
Yeah, that might have been a remnant of the video. But once you release them, and they come into detection range of 3 receivers, you get a triangulated position of your sea turtle. So once they come into that square of receivers, then you get a point every time you get a detection. So because they were only interested in the first few hundreds of meters of these animals moving away from the beaches, you use certain programming that you get a detection every couple of seconds or even less. You can have like 1.2 seconds, for example, that you get a signal. It's then, well, it's a bit technical, but you can have shorter signal delays, so you get more signals out of it. And then the shorter the signal delay is, the more positions you get. So it's not you could make it a line as well, but then you would smooth the track a little bit.
Thanks,
right?
Thanks very much. Let me rephrase or revisit. Am I more likely to lose fish when they are detections of fish when they are shallow, or when they go deeper. So
yeah, okay.
In shallow water, especially, I'm shallow water, maximum 20, 30 meters depth, 40 meters depth. I have a network of stuff like that.
Yeah, well, losing due to predation, you mean? Or?
No, just by physically changing depth because of propagation. Like in very shallow waters, I'm likely to not hear them.
In general, yeah, okay. In general, coastal areas, or let's say shallow areas, are noisier.
Yeah, that's why I'm...
Wave action, precipitation of heavy rain, moving particles,
sounds,
whatever it is. Usually the open ocean is quieter. But again, that depends on, weather patterns and whatnot. Usually the shallower it is, the noisier it is.
And that's why you discuss for many control studies, range detection.
Yeah, that's what you want to do.
Preliminary work.
If you do, for example, have an array that stretches from shallow waters, let's say 5 meter depth to 500 meter depth, and you have increments of a few hundred meters or kilometers or whatever, you've got a receiver. I would do a detection range, let's say in shallow waters, mid-water, and then in the deep water. But because the detection ranges might be completely different. Right? If you've got different mooring designs, same again, shallow water might have a different mooring design, very easy, diveable, maybe only a stick and the receiver is attached to it. If you've got different mooring designs, you want to do different range testing as well with all of the different mooring designs that you have, because not every mooring is static and might not emit any noise. But if you have got shackles, for example, moving parts on your mooring design, they might generate noise and they might interfere with the detection range that you get out of it.
Okay, thank you.
Yes, please. Yep. It depends on how many receivers you have. It depends on what kind of question you want to answer. Are you interested in the movement only in and out of the estuary, for example, in the open ocean? Then you only need a gate, for example, or a double gate if you want to see directional movement. If you've got a single gate, you want to see movement, let's say, from the estuary outside of the estuary. Then you get presence, absence along the strait. If you want to see directional movement, you can have a double gate, for example, first detections on the first gate, last detections on the other side, on the outside, and then you see animals are moving out of the estuary or inside of the estuary again. And then it depends on your question. Do you want to have a look at habitat use, distribution, range, or whatever of your animals within the estuary? Are you interested in coastal movement? And that's where you strategically choose locations, receiver locations, and moorings that make sense to answer your questions the best way you can with the budget that you have, right? You might have 5 receivers or you might have 50 receivers, right? And that gives you a certain resolution. Yeah.
When a tag is placed subthermally, is there any chance of rejection by the fish's immune system?
It can. With gravid females, for example, if they do have a lot of eggs inside their body, in certain species, there have been occasions where a transmitter that was close to the oviduct, for example, was being ejected. Other species can eject a transmitter, for example, octopus, very smart animals. There have been occasions, for example, where octopus have been tagged externally. They just rip it off because they didn't like it. Then they were tagged internally, so they take one of their arms, go into the body cavity, and rip it out from the inside and discard of it. Subcutaneously, it's usually not done because they're usually going into the coelomic cavity of an animal. So you make the incision, and then it stays between the organs and the muscular tissue, basically. And then tag retention is very good, really. Some occasions, some species, there's evidence that it has been ejected, but usually the retention rate is very, very high. External tagging, different story. That depends on your application, how well you attached your tag on the animal, the behavior of the animal. Is it going into like under reef structures, rocks where the transmitter can be rubbed off? Do they have inter-animal behavior where they're rubbing against each other, fights, mating, whatever it is? Then tags can come off. Yeah, but tag retention internally is usually pretty good, very good actually, and externally it depends.
I was talking more about the immune system response rather than the behavioral.
Yeah, okay, I see. Yeah, no, they're made of Delrin and epoxy, and it's a very inert, very sturdy component that is usually not getting infected or being rejected by the animal. No, there has been, I don't know of any evidence of that. Okay. It can be that the only thing that I can think of right away is that you, for example, if you've got a tag that has got 2 attachment points and you want to have it at the dorsal fin of a shark, for example, if it's a juvenile shark and it has got a big growth rate or great growth spurts, the dorsal fin will grow as well. If you've got 2 attachment points, then they can rip out, for example. For juvenile sharks, then you use only 1 attachment point because then the tags cannot rip out, for example. But that's kind of it's not a rejection as such, but it's one of the possibilities how we can injure an animal.
Right, exciting stuff. Thank you very much. But I really appreciate the whole presentation. The presentation was great. You know,
thank you very much.
That's my personal taste. Thank you. And thanks very much. The booth is open.
Yeah, absolutely.
You're gonna be here till Saturday lunchtime. Thank you very much. And sorry to be scared. I always do it. You know?
Thank you.
This was my last slide in Volos. Our project proposal had just accepted our National Research Council. We have 3 work packages in this project. First one, population density of 2 important sea cucumber species. And second work package is related to reproduction cycle, which my colleague and my wife already presented session.
You are not speaking at home. You're discussing in the conferences.
Yes, conference. Yes. And last work package is related to improve product quality. We add these work packages because our fishermen collect sea cucumbers and put plastic bags and in seawater, start from morning, all the day, all product in seawater. They lose quality. We add these work packages to the project. My work packages is the aim of this work package is to compare the population density between exploited and non-exploited fishing area. In the figure, the upper part, there is a Çandarlı Bay, open the sea cucumber fisheries, and lower part, lower part, İzmir Bay, close to sea cucumber fisheries. Methodology was very simple. We sampled seasonally between September 2018 and March 2020. And we have 2 bays, 2 stations in each bay. And each station we have 2 depth ranges. Shallow, 0 to 10 meters, and deeper, 10 to 20 meters. And we have 6 replicates in shallow stations and 3 replicates deeper station. Why we choose this? Because in preliminary study, we saw the most of the individuals prefer the shallow habitats, like Posidonia, sandy, and muddy area. The deeper waters, no any individuals, mostly. We also asked 2 fishermen, and then we prepare this kind of sampling strategy. We use underwater visual sensors technique. In this technique, first, we use 100 meters line, light, the sea bottom, randomly. And then there's 2 buoys each end. And we record geography position of each buoys. And 2 divers using the both sides of the line, and each diver responsible 2 meters wide and 100 meters long, the area they collect data. This means in each replicate, we scan 400 square meters sampling area. This means there are a lot of ropes, buoys, or crane placing them for us and record the GPS locations. And then we dive. There is a video, but you see this is only one diver because another diver take a picture. First, we diver when to encounter sample, they record the depth, habitat, and collect the individuals to the back and bring to research vessel. And this is the result. The left part of the tables, you see the seasons, and another depth counter, shallow and deeper, shallow, deeper, and green Denizköy and Şakran stations open the sea cucumber fisheries, and Urla and Güzelbahçe in İzmir Bay, red ones. This is the close to fisheries. In the table, you see mean density of the Holoturia tubulosa. Red circle means we couldn't find any individuals. The whole huge area, zero, no individuals. And the green ones mean highest diversity. Highest diversity, 0.006 individuals per square meters. Almost nothing. And the same story in closed area. Urla and Gülbahçe in İzmir Bay, it's closed for cucumber fisheries. The highest mean density, 0.04. And a lot of red circle. And Holoturia story is very, very bad. You see a lot of circle in fishery, open the fisheries, zero individuals. And also the same in İzmir Bay, open the fisheries. And I like this table. If we compare these findings, this or mean density, please look first column. Colon and Jacques 1993. The density 3.8 individuals square meters. The date is very important. This paper published in 1993. This means before sea cucumber fisheries started in Mediterranean, because this fisheries started to 1996. This means that time, how to say, natural density near the 4 individuals per square meters. And then my colleague from Greece found Pakasitos Bay 0.01 individuals per square meters. And other Turkish guys. And last one, this study, 0.002. You see the very rare to population density. And this is our export volumes, belongs to last 10 years. Before we have export this date, but not good statistical data. And good data start to 2014. And increase until 2019, which our project finalized. We analyze data, and situation is not good, but production highest level. And that time, our General Director of Fisheries Management put the quota. Quota application started. Quota gave 18 different company to 2,500 tons. But you see, the highest level, we just produced 1,200 kilograms export, double. They put the quota double. And then story start to collapse. You see, now last year, we just export 2 tons from 1,200 tons to 2 tons. Yes, this is the story. Conclusion. Why did stocks decrease so much? This is what use of many fisheries management tools. Many fisheries management tools, I mean, we have closed season, closed area, boat certification, diver limitation, only daytime fisheries, a lot of good management on the paper. But the problem is, first one, illegal fishing could not be prevented. From northern Aegean to South Aegean coast of Turkey, everywhere, every young diver collect illegally sea cucumber because of money. one day, one young people can how to say, get money, 1,000 euro one night. In this reason, illegal fishing, boom. And second, bad governance, the quota application. Because for quota application, you must know the stock. If you don't know stock, you cannot calculate quota. And then you see this, maybe you can put 1,200 kilograms quota, okay? But the government put double. In this reason, now we are only 2,000 sea cucumber export last year. This year, I have a lot of students. They do this job. They out of these fisheries because they couldn't find any individuals to sell to processing fabrics. That's all. Thank you very much, Chairman.
Thank you very much for this excellent presentation. If there is any question also on that? Professor Gansis from
yeah, yes, sir.
It's very, very nice and very interesting because we are starting to learn the value and create fisheries. So, is there any possibility that the collapse of some populations in some cases could be related to the macroalgae? The presence of sea plants in some places.
Not directly, but you know the sea cucumber has very big capacity to how to say, handle the sediment, to remove the organic matter, and at the same time, you know, the Pinna nobilis also must dead, and sea cucumber now or shore, no sea cucumber, no Pinna nobilis. This means no any cleaning the water or organic matter. Is some places visibility very low now because we don't, the animal not remove the organic or inorganic matters. Now, very big, the underwater, I start to dive 1986. And I know that time, but now a kind of become a desert, some places. In our station, we don't have any station, sampling station near the sea cages, but my old observation, yes, there was a lot of sea cucumber, but the fisherman knows very well and collects all of them. Yes.
Fishermen were informed that send this abroad because we don't
yes, China. Export to China. All of them.
There is a bad experience. You have to take care because
yes.
Even species that are considered not acceptable. Told me that the factories are not. They don't care at all if
yes.
ask for another single.
Yes. Yes. In actually, you know, the Pacific species very big. Our species very tiny. But the Chinese people likes very much. Low price, but they collect all species. Octopus? No.
Kind of pots, no?
Pots?
How do you collect?
For octopus or sea cucumbers? By hand, scuba diving, by hand. Yes, it's very, very easy. Why this fisherman can collect 1,000 euro per night? Very easy to collect. Of course.
I saw the graph with the exports. The graph with the exports.
Yes.
But is the only data available that we have regarding because the export data is not obviously the catch data from seacucumber, right? So there are some quantities of seacucumber must be leading to the Turkish market.
No, we don't consume all of them to export. Also, all quota gives the processing fabrics. The government collect data from this one. Only summertime, some fishermen use only bait, but very few. Very few.
Because you said that you mentioned that 25 years ago that you were dying,
yes.
You saw a lot of seacucumbers.
A lot of.
But because we are living in an ecosystem-based approach, so do you think that apart from illegal fishery, touristic activities, the expansion of tourism in Turkey during the last 10 years is huge. So is there any connection with the huge decline of seacucumbers with the expansion of touristic activities? I said that in order to study also some abiotic factors, or as Yannis says, about to examine other parameters that are impacted on seacucumber.
Yes, of course, there is some interaction between the abiotic and biotic factors, but tourism sector just, how to say, in these two stations, for example, especially Chandarlı Bay, the open to seacucumber fisheries, we cannot say very high people density or tourism activity. Not too much, but mostly secondary house in the summertime become crowded, and most of the time this house empty, not mostly populate the human activities.
Okay, thank you very much.
Oh.
Okay, proceed to the next speaker, which
Ladies and gentlemen, before I start my presentation, I would like to express my special thanks to organizing committee because as far as I saw, it's been a great scientific event. So once again, thanks for your efforts, both the scientific committee and organizing committee. So as you can see from the title that it's a collaborative article, me, Ozan Soykan, is the presenter, and my colleagues are from Muğla Sıtkı Koçman University and from HydroMediT University. So the fish's biological characteristics are important values for the stock assessment studies. Growth parameters provide some indication of resource utilization and the effectiveness of management strategies. When age and growth studies are evaluated in combination, it may be easier to understand the relationship between population size and biomass. This understanding is the basis of modern fisheries resource allocation and management, and fisheries management should be designed based on biological data to understand the status of managed fish stocks, especially the growth parameters L infinity, K, and T0 are used in almost all stock assessment models. However, the studies have mostly focused on commercial species. Even if decline in fishery resources is a different topic, ecosystems should be considered as a whole. Therefore, either commercial or non-commercial species should not be considered different components of the fishery resources. During the brainstorming phase of this study, we thought, all right, how many age growth and mortality studies have been conducted up to 2023? What are the details of these studies in the Mediterranean region? And the final question is, gathering information about the Mediterranean fish species could be helpful for the fisheries scientists. Thus, this review came out to deeply learn about age and growth studies in the Mediterranean and Black Sea basins. The obtaining results are considered to be very important in terms of showing neglected species of fish biology. As you can see on the slide, the Mediterranean was separated into subregions by FAO as Western Mediterranean, Central Mediterranean, Eastern Mediterranean, and Black Sea. Regarding the methodology, Mediterranean fish species were determined from the fish-based catalog. Age growth and natural mortality values of all Mediterranean fish species were taken based on fish-based website. And also, during this process, an artificial intelligence-powered research tool, Semantic Scholar, was used. Studies were also researched on Google Scholar to keep sight of any study. The cited literature in the obtained papers were also controlled and checked. You can see a screenshot of what we have done during the data collection phase. And we categorize the data. According to the results obtained from the literature search, fish species have age, growth, and mortality studies that have conducted in the Mediterranean were selected and classified. Next, a detailed analysis was conducted to learn about the Mediterranean studies, such as regional, species-specific, number of studies, number of families, and etc. Species threatened levels were also determined by the IUCN Red List. So here we come to the result. According to the fish base, the Mediterranean basin includes 778 fish species. In this number, 557 are native, 135 are introduced, 47 are endemic, 23 species are questionable, 14 are misidentified, and 2 are error in name, or in other words, incorrect spelling. All species without native and endemic species were excluded from the data set. So totally, we considered 604 species. All age, growth, and mortality studies were conducted in 22 different countries in the Mediterranean. The first 3 countries that have AGMS, which means age, growth, and mortality studies, briefly, are Turkey, Italy, and Greece. A total of 796 studies belong to 185 fish species have age, growth, and mortality results or information. In some papers, while one species was studied, on the other papers, or the rest, include 2 or more than 2 studies. And this map was built according to species numbers among those studies. In this slide, you can see the number of studies with the timeline. So we can say that the number of studies had started to slightly increase by the late 1940s, and then we have a sharp increase in the 1980s. The selected 185 fish species belonging to 72 families were 161 of Osteichthyes and 24 of Chondrichthyes. So it means that majority belong to the Osteichthyes. On the other hand, in the Mediterranean age and growth studies, we also showed a dominancy in favor of Osteichthyes, which is more than 95%. With regard to family-based distribution, the first 10 families and species, as you can see from the slide, that's Sparidae, Mulidae, and Mugilidae. Sparidae includes more than 170. Mulidae includes almost 90, and Mugilidae family almost 70 species respectively. Regarding the Sparidae family, we see that Boops, Pagellus erythrinus, and Speccara simaris are the most studied species. And regarding the Mulidae and Mugilidae families, Mulus barbatus and Mugilocephalus are the other most studied species. Regardless of the family-based distribution, we see that Meliscius is the leading fish ever studied in the Mediterranean. And also we considered the commercial importance of the species, and we see that vast majority of the species have commercial importance. We also categorize the species according to their endangered levels. So here we followed IUCN Red List status. As you can see from the slide, most of the species are in the least concerned status. And we have also nearly 4% of critically endangered species. And in this slide, you can see the critically endangered species. These are Anguilla, Spencer, Huso, Rhinabatos. Species are among these species. And before discussion, I should add that among 160 commercial species, we see that 31 fish species encounter existence problems, and 15 of these 31 species are unfortunately belong to Chondrichthyes family, or classes, sorry. The Mediterranean Sea has high species richness. The number of native and endemic species constitutes considerable amounts of total fish species in the Mediterranean. The Mediterranean seawater temperature is unfortunately 1.4 degrees above than before. Therefore, an increase in the Mediterranean temperature would lead to an increase and to northward spread of introduced species. Thus, this phenomenon leads to an increased species number in the Mediterranean through the invasion by introduced species. Turkey, Italy, and Greece are big peninsulas, and all the shores of these countries are located in the Mediterranean and Black Sea regions. On the other hand, France, Spain, and Morocco have coasts along the Atlantic and Mediterranean, and studies in these countries are also relatively high. But in this study, only Mediterranean studies were considered and sorted. Therefore, study numbers of Turkey, Italy, and Greece are higher in other countries in the Mediterranean and Black Sea regions. And we can see that these three countries are dominating the fish biology studies in our basin. The number of studies has increased since 1980s. Especially in 1980s, researchers tend to learn ecological parameters of the exploited populations with direct methods. In addition to this, technical progress has led to an increase in the quality and efficiency of fishing gear. For example, the catchability of Nepros norvegicus increased with technological advances. It's mentioned in many literature. That's why I tell it to you. In this finding, it can be said that an increase in the number of fisheries studies in the Mediterranean is attributed to technological advances in fisheries, such as spread of electronic devices, high power engine use, and technological improvements of the fishery in general. Logically, the increase in the fishing capacity and fishing ability has made it easier to reach the desired species and to do the sampling for the species of the Mediterranean. A few seconds ago, I mentioned about sampling, so now I will talk about the sampling procedure. According to numbers, most of the species are bony fish. In contrast, 24 cartilaginous fish species have been studied. Cartilaginous fish are known mostly as predators, and they are situated at high levels of the food chain. Therefore, they are rarer when compared to other fish species. The lesser studies in cartilaginous fish may be attributed to reaching these species because it's difficult and being lower abundance in the catch composition. Furthermore, fishing techniques may be effective in capturing cartilaginous fish species. In this study, cartilaginous fish species mean depth range were minimum 21 meters and maximum 821 meters. So these 21 species occur at depths above 200 meters. As it is known, deep zone studies or deeper zone studies require higher fishing equipment and research vehicles. In our opinion, the most limiting factor for cartilaginous fish species may be the depth. On the other hand, most of the cartilaginous species are on the IUCN Red List. This situation may be also another reason for the low number of studies. Again, regarding the sampling, Sparidae is the most studied family in the Mediterranean basin. Spirit species are commonly found along the shores. Most of the spirit species are caught by different fishing gears. For example, boops is captured by different fishing gears such as trawl, purse seine, and handline. On the other hand, Mulidae species are captured mainly by trawl nets and also by gill nets. Furthermore, even though Mulidae species are captured by many fishing gears, their mass capture is from traps, especially during their migration. Therefore, sampling of Sparidae, Mulidae, and Mulidae species is relatively easy when they compare with other species. There is a difference between doing sampling and samples coming to you. Behaviors such as being a school, being in a species-specific habitat, and seasonal migration may make the sampling easier. For example, in this slide, I would like to show you a bottom trawl holeback. How can I forward the video? Is it by scrolling or by clicking on the mouse? I think video is not functioning. Anyway, I'm sure most of you are familiar with a trawl holeback situation. So sometimes you can even collect a Sparidae family by a handline. But when you are working with deep sea fishes, for example, Helicolenus dactyleptorus is one of those. And at the end of this presentation or this video, unfortunately, there was an individual of Helicolenus, but we cannot function the video. So when you go deeper zones, it's really difficult to sample what you want. And in this part, I want to talk about the cartilaginous fish species. According to the fish base, there are 86 cartilaginous fish species in the Mediterranean, and 68 of them are under risk. Study results show that 24 cartilaginous fish species have been studied in terms of age and growth. Among these species, 15 are or of commercial importance. On the other hand, GFCM statistics show that there's a decline in chondrites capture in the Mediterranean. Some of the researchers mentioned that more than 98% of the shark catch weights have been in decline since the last 200 years. And if the situation continues like this, the extinctions will be inevitable. The loss of top predators could lead to serious ecological effects. Elasmobranchs are generally caught incidentally, and bycatch and direct fisheries on cartilaginous fish species are not very well monitored in the Mediterranean. Therefore, the biological parameters of cartilaginous fish stocks should be identified for both ecological and commercial importance of the elasmobranch fishery. In conclusion, most of the species in the Mediterranean have no age and growth studies, neither age nor growth. Biological identification of the fish stocks serves not only commercial sustainability, but also ecological sustainability. In this sense, the construction of a reliable food web is important. Therefore, non-commercial species should also be taken into consideration in terms of age and growth studies. I would like to thank you for your attendance. Thank you very much. You're welcome. You're welcome.
Any question?
Then nobody understood what I said.
Let's start.
Okay, just.
You were part of that book then. Yeah. I try to find it. This one.
Study or it's a cycle study because of the size of the floor.
Yeah. It's a good point. This is a commercial vessel, but when you look at the mesh size, it doesn't look like a commercial vessel because mesh sizes are really small. But this is the general attitude of Turkish troll fishermen. They put a cover, or how can I say it? Yes. They put a cover just out of the codent to avoid fish catches. So this is a important point. Thank you. Mm-hmm. Yes, no. Thank you. I didn't mention that, but as far as I know, I don't have, or I don't remember the specific numbers of the preserved fishes, especially regarding the cartilaginous fishes. I can say that in recent years, the number of preserved or conserved cartilaginous number of fishes are increasing. So that's a good point of the Turkish fishery management. Yeah, actually. Yeah. Yeah, I know. Yeah. Mm-hmm.
Apart from my European.
Yeah. Yeah, of course. Yeah. You are exactly correct. It depends on the timing or season of the troll fishery. For example, it was a summer fishery. So during the summer fishery, the trolls are allowed to perform at international waters. So during the international waters fishery, holding time may take up to 7 hours sometimes. Yeah. But during the normal fishery time, normal the legal time, it's 2 hours, 2.5 hours. Thank you.
From Skaya, right?
Hello everyone, I'm Simge Bozkaya and I'm a Master of Science student at Ege University's Faculty of Fisheries in Turkey. Today I will be presenting on the relationship between eye diameter values, total length, and head length in some species of the order Signatiformes. Fish species exhibit behaviors related to sight, such as searching for food, evading predators, looking for a nest, or mating. In her 1996 book on Ichthyology, Demir from Istanbul University stated, structurally, the fish eye is similar to that other vertebrates. The eye connected to the brain via the optic nerve, and its lens consisting of 3 layers known as sclera, choroid, and retina, as you see in the figure. Fish vision differs based on their habitat conditions, especially between light and dark zones. Also, the depths affect the fish vision. For instance, the light levels in the mesopelagic region allow certain fish species to detect potential prey. The aim of our study is to examine the relationship between eye diameter and total length in Signatiformes species. In our research, we examined 573 specimens within the Signatiformes order. These species are Signatus abaster, Signatus acus, Signatus schmidti, Signatus tenerostris, Signatus tifle, Signatus vargiatus, Nerophis ophidian, and Fistularia petimba. The signated samples we examined were gathered from the coastal regions between 2018 and 2022. Eye diameter values were determined as a percentage considering their relationship with total length and head length. In addition, we investigated the correlations between eye diameter measurements, total length, and head length. We applied the regression analysis to assess the relationship between eye diameter and total length and a t-test to identify any significant differences in eye diameter across genders. Signatus abaster holds the distinction for possessing the smallest average eye diameter in relationship to both head length and total length. In terms of head length, the average eye diameter of Signatus abaster is relatively small, with a mean and standard error of approximately 1 millimeter to percentage of head length. When considering the total length of the creature, the eye diameter maintains its small proportion, coming in at approximately 9 millimeter to the percentage of the total length. This suggests a unique adaptation of this species where it has evolved to function with relatively smaller eyes compared to the overall size of its body. In this comparison, there is a notable contrast between Signatus abaster and Fistularia petimba. Specifically, Fistularia petimba stands out because it displays the largest mean and standard error values. These values are significant with the head length percentage approximately 92 millimeter and a total length with filament percentage of just 2.9 millimeter. Therefore, this comparison highlights the unique features and proportions of Fistularia petimba in comparison to Signatus abaster. A sex-related difference in eye diameter values was observed in only one species, as shown in Table 3. Specifically, a significant difference in eye diameters between male and female Fistularia petimba specimens was noted. Female Fistularia petimba specimens were found to have larger eye diameters than males. Table 4 indicates a notable regression relationship between total length and eye diameters in the species of Signatus acus, Signatus tenerostris, Signatus tifle, Signatus vargiatus, Nerophis ophidian, and Fistularia petimba. The nature of this relationship seems to be interconnected with these species' phylogenetic structures. We conducted a study to explore the differences in eye size within particular Signatiformes species and between sexes. According to Walsh, visual ability is closely related to size of the eye. Fishelson in 2004 noted that species active during the day typically have eye diameters that make up roughly 10% of their total length. In contrast, species active at night tend to have slightly bigger eye diameters, taking up about 12 and 13% of their total length. Our results for Signatiformes species correspond with these proportions, prompting us to categorize these species as diurnal. Laughlin suggested that increasing eye size can enhance energetic properties, especially eye mobility. The bentopelagic fish species like Fistularia petimba support this theory as mentioned by Mundy. Lundstedt put forth the idea that a reduction in eye size could potentially be an evolutionary adaptation to lower the risk of predation. It's worth noting that natural habitats like seagrasses, which are home to cryptic species such as the pipefish, might offer adaptive advantages by providing ideal hiding spots and increasing the likelihood of successful prey capture. Moreover, differences in eye diameters between sexes were noted in some species. In particular, within the Fistularia petimba species were noted that females tended to have larger eye diameters. This could be a result of natural selection promoting sexual variation in the head region among fish species. In conclusion, this research is a pioneer in determining the eye diameters within the Signatiformes order, particularly for pipefishes that inhabit seagrass beds and are active during the day. The smaller eye diameter plays a dual role. It not only shapes feeding routines, but also serves as an advantageous evolutionary trait, reducing in their visibility to predators. I appreciate your patience and participation. Thank you so much.
Any question? Yes.
Table 4. Yes, yes. Okay. We examined total 573 specimens, but these consisted 7 from Signatus abaster, 235 from Signatus acus, 9 from Signatus schmidti, 23 from Signatus tenerostris, 99 from Signatus tifle, 11 Signatus vargiatus, 39 from Neurophysophitinan, Fistularia petimba is 50. Yes. So actually, I can say that all of these calculated from my supervisor, so I can ask. Okay, thank you so much. Thank you.
If you want to help study this relation between the eye diameter and length or mouth dimension, it's not obviously to be linear. We can have other forms of equations. So maybe we can check another option.
Yes.
Actually, also in nature, there's no linear relations. Yeah, as a human, our brain is to transform to linear and linear equation. But in your case, maybe you can check also other form of equations, polynomial, some exponential, maybe because you have a dimension of eye. It's not linear like change so much. It's not like if you check your data,
yes.
Scattered load, you may see a pattern like a line, but not linear, like non-linear as horizontal.
Yeah. Thank you so much.
Hi, everybody.
You can explain to us why jet metal research,
Yes.
how this belong to fisheries sector?
I don't included fisheries section, but I see my presentation in fisheries, include fisheries. My name is Murat Yabanlı. I am professor from Muğla Sıtkı Koçman University, Turkey. Before I start my presentation, I would like to thank you everyone who contributed to bring us together in such a nice organization. My field of study focused on two keywords. First one is heavy metals and others is microplastics. In my presentation, I will present the mercury, cadmium, lead concentrations in the muscle tissues of soulfish caught from the Güllük Bay, southeast of Aegean Sea. Heavy metals are among the most harmful pollutants for sea and living organisms. Heavy metals are a group of metals and metalloids that have relatively high density and are toxic even at ppb levels. For example, cadmium. Among metals, cadmium, lead, and mercury are hazardous pollutants referred to as the toxic trio because they cause adverse effect in plants and animals even at low concentrations. ATSDR substances list you can see on this slide. Three of the list of the 20 substances are mercury, lead, and cadmium that subject of this study. Bioaccumulation of harmful elements in fish occurs either directly from the water or through trophic transfer prey. Ultimately, these toxic elements reach human through the diet. The saying big fish eat small fish becomes very meaningful in this point. Aim of the study, one, the concentration of mercury, cadmium, and lead elements in the muscle tissues of common soulfish traveled from Güllük Bay, which is one of the main important soul fishing areas in Turkey, were determined. Second, the relation between sex, total length, and weight of soulfish and heavy metal concentrations in muscle tissue was investigated. And last one, consumer health risk assessments were carried out in terms of studied elements. Güllük Bay is a point under the pressure of pollutants. If we say a few of them, a point close to the airport, highways and port, intensive tourism activity, agriculture, and livestock activities. In this slide, I wanted to show you the process of determination of toxic trio. First step, sampling, and then dissection, microwave wet digestion using nitric acid, and determination of concentration heavy metal by ICP-MS. We got the data, then what did we do? We determined the relationship between the elemental concentrations and the biometric characteristic of the fish. And We analyzed risk assessment for consumer health. These consist of estimated daily intake, carcinogenic risk, hazard index, and THQ. In this slide, we seen biometric trace of saltfish. The female to male ratio 5:6, mean maximum weight 204 grams, mean maximum length was 27 centimeters. Mean element levels determined in muscle tissue were found lower than the codex limits. Codex limits 0.05 ppm for cadmium, 0.30 ppm for lead, 0.50 ppm for mercury. On the 11th slide, you can see the results of consumer health risk assessments in terms of four parameters, which are estimated daily intake, carcinogenic risk, hazard index, and THQ. We can say clearly consumption of saltfish is safe for consumer, include children and adults, in view of mercury, lead, and cadmium. This slide also contains some of our findings. What are these? There was a strong positive correlation between cadmium and lead concentration and total length and weight. There was weak positive correlation between mercury and total length and weight. There was weak negative correlation between sex and toxic trio concentration. There are no adverse effects for children and adults in terms of mercury, cadmium, and lead elements associated with the consumption of common salt. Aquatic ecosystems are highly dynamic and under pressure from pollutants. For this reason, it is very important that risk assessments are made periodically in terms of contaminants regarding consumer health in seafood. Thank you very much. I tried to make a presentation without going into too much detail. I hope you enjoyed my presentation. Thank you very much for listening to me. Efaristo. Yeah. We wool muscle tissue for study. We are study diet muscle tissue. For this reason, all muscle tissue homogenized and then determined heavy metal concentration in muscle tissue. All muscles. Yeah, yeah. Thank you.
Towards the end that there was no consumption. This is based on legislation, basically, based on law, on minimum concentration of
we use mean concentration, all calculations, and most important point, consumption rate. Consumption rate for Turkey, 6 kilogram annual year. Yeah.
If case the consumption will increase, we'll have a problem.
Our levels very low because no problem for Turkey.
Don't like to eat fish too much.
Yes.
Thank you.
The next also is the same. Metabolic. Something pieces the after this, and then for that.
Yeah, yeah. Thank you very much. My name is Dr. Ioannis Georgoulis, and I will present you our research about the energy metabolism rearrangement that enhance the heat tolerance of the heat-hardened muscle, Mytilus gallobronchitialis. First of all, the sea surface temperature is highly increased in the last decades in the Mediterranean Sea and especially in the eastern part. This increase of the sea temperature will affect the molecular, biochemical, and physiological level of organisms, but it affects not only the individuals, but also higher level of organization like biographical and population level, and eventually can affect the whole ecosystem. According to the hypothesis of oxygen capacity limited thermal tolerance, it is known that the increase of the temperature can decrease the intake ability and the transfer of oxygen to tissues. That will decrease the aerobic capacity and activate the anaerobic metabolism. As we can see from this diagram, ectotherms live in a range of temperature where they demonstrate their maximum aerobic capacity. Beyond these limits, there start to appear biochemical, phenotypical, and behavioral changes, while in even more extreme temperatures, beyond the critical temperatures, we have heat shock gene expression, increase of oxidative damage, and reactive oxygen species production. While even higher temperature, beyond the denaturation temperatures, we have increased damage of proteins, cell degradation, and limited survival. The question is, have the organisms the ability to enhance their thermal tolerance under specific thermal conditions? According to the German philosopher Nietzsche, that which does not kill us makes us stronger. According to the Hormesis, the exposure to low amounts of a stressor can increase the organism resistant to future higher doses of the same stressor. As we can see from this diagram, a low intensity of a stressor could be beneficial for the organism, while the higher intensity of a stressor could be harmful and increase the damage to the organism. A very good example is the myth of Mithridates that sought to harden himself against poisoning. So what's happening during the hormesis? When an organism is exposed for the first time in a mild or a sublethal temperature, there are some hormetic responses like increase of autophagy, increase of unfolding protein response, and heat shock protein response that will create a preconditioned state. This state will help the organism where when it will exposed in a future intense stress, it will be, we will see higher stress resistant and cell survival. On the other hand, if the organism will not create this preconditioned state and is exposed for the first time in this intense stress, then we will have increased cellular damage and cell death. These hormetic responses will help the organism in order to acquire stress memory that help it for a faster and more efficient response against this intense stress. We saw in our title, we use the term of heat hardening. The heat hardening is defined as a rapid beneficial hormetic response that improves the heat tolerance after a brief exposure to sublethal temperatures. During the heat hardening, there is a coactivation of multiple stress signaling pathways that lead to phenotypes with increased resistance. So the aims of our study was to investigate the ability of the Mediterranean mussel Mytilus gallophryngialis to enhance its thermal tolerance to increasing temperatures after it has been subjected to the heat hardening. So we want to give answers of which are the possible adaptations of biochemical pathways involved in energy metabolism that benefit this phenomenon. For our methodology, we used some mussels. Our methodology is divided in two phases. The first one, as we can see, is the hardening phase, and the part of mussels that we collected from local aquaculture were transferred to our laboratory and placed in this type or this system of tanks, where we can easily increase and decrease the temperature. The hardening phase, the hardening protocol, is based on the Hudson's repeated critical thermal maxima with minor modifications. At first, we start to increase the temperature at 18 degrees to 27 sublethal temperature for the Mytilus gallophryngialis, and mussel remained in this temperature for 2.5 hours. The increase was fast enough with an increase rate of 1.5 degrees per minute. After they stayed to 27, the temperature were decreased again back to 80 degrees. They remained for 22 hours in order to recover. And the same procedure were repeated 3 more times. The mussels that have were subjected to the hardening phase were named as hardened mussels, while another group that we don't use them for the hardening phase were named as non-hardened mussels. So for the second phase and the main part of our methodology, we have the acclimation phase, where in new tanks, 4 new tanks, both non-hardened and hardened mussel were placed, and we increased the temperature to the 3 of the 4 tanks to 24, 26, and 28 respectively, while the 4th tank remained at 18 Celsius in order and were used as control mussels. Mussels stayed in these elevated temperatures for 20 days, and samples were taken of mantle tissue on day 0, 1, 5, 10, and 15. For our experimental analysis, we analyzed some metabolites using the metabolomic analysis with NMR spectroscopy, and that took place in the Alfred Weninger Institute in Bremerhaven, Germany. And also some these 2 genes, PK and PepsiK, RNA expressions using real-time PCR. In our first results, we will see 2 different metabolic strategies between a non-hardened and hardened mussel. First of all, the non-hardened mussel, we see that there's a depletion of their adenylated pool of ATP plus ADP. That was significant lower compared not only with the control group of 18 degrees, but also with the hardened mussels. Also, we have accumulation of hypoxanthine levels, especially in 26 and 28 degrees. And the oxidation of hypoxanthine can generate reactive oxygen species and increase the oxidative damage. It seems that the non-hardened mussels use the purine breakdown pathway, decreasing the adenylated pool and increasing the hypoxanthine as a final product. On the other hand, the hardened mussels have a completely different, we can say, strategy. First of all, we have increased levels of inosine. Compared to the non-hardened mussel, we see completely no increase of inosine. And also decreased levels of hypoxanthine, also compared to the control group and the non-hardened mussel that were increased. This shows that there might be an inhibition of the previous strategy of the purine breakdown pathway that could be beneficial for the heat tolerance and the hypoxia survival. It seems that in the hardened muscle might have a different strategy and use the purine salvage pathway that help them to generate their ATP production and showing, as we can see here, higher levels of adenylated pool of ATP plus ADP. The adenosine and inosine can provide an alternative source of energy and prevent the inhibition of glycolysis and Krebs cycles in the hardened muscles. Also, another metabolic strategy we can see in these three metabolites of acetate, fumarate, and succinate. In the first one, the hardened muscles, we have no accumulation of either of these three metabolites, and it seems that they use a more robust aerobic pathways like the TCA cycle here, or even they have a dilation of their of the switch to the anaerobic succinate metabolism. On the other hand, we have accumulation of all these three metabolites in the non-hardened muscle, the blue ones, showing an insufficient oxygen supply and an onset to the anaerobic succinate metabolism, as we can see in this, using this pathway. And these results come in line with the mRNA expressions of PK and PepsiK. And as we can see, the non-hardened muscles will have lower levels compared to control of PK mRNA expression that show the decrease of aerobic metabolism, as we saw before, and also increased level of PepsiK mRNA expressions that channels glycolytic products towards the anaerobic succinate production, as we said before. On the other hand, the hardened muscle have both PK and PepsiK. Both of these genes were transcriptionally upregulated, showing the upregulation of aerobic pathway through the TCA cycle and the oxidative phosphorylation. But also, we might have an increased, it seems that they might be prepared also for the use of the anaerobic metabolic pathways in order to supplement their ATP production, their energy supply. So in order to conclude, it seems that the heat hardening, during the heat hardening, we have a metabolic reorganization that helps the organism to increase their production of ATP through an enhanced TCA cycle and oxidative phosphorylation, and also the use also of the anaerobic metabolic pathways as a supplement. All this energy, the increased energy, will be helpful for the organism in order to increase their protective mechanism against the oxidative stress, like heat shock protein response, antioxidant defense, osmoprotection, and autophagy, mechanisms that we have already seen that they are upregulated in other research. So all these mechanisms, they are energy demanding, and this increase of supply will help to will help the organism against the oxidative stress, increasing their thermal tolerance and the survival of heat-hardened muscles. So the question is, can we use the hardening process in the field? In fisheries or aquaculture, can we provide lower mortalities for mussels with higher thermal tolerance? Can we take better quality of product, or even have mussels a better response against other stressors, not only the temperature? So more field experiments will be needed. So this was our presentation, and here you can find more information with many other metabolites that's showing this increase of thermal tolerance. Thank you very much for your time. Yeah, the last one is the general. Yeah. So we know that already the climate change had caused many problems, not only in aquaculture but also in fisheries, with many mortalities of many organisms like mussels that they cannot move; they stay in one place. Already, the sea surface temperature have been near 28 degrees for over 10 days, maybe during the summer season in the last 3 years. So in aquaculture field, for example, they have been estimated over 90% of mortalities. So what we want to achieve, if we can use this hardening process in order to increase the thermal tolerance of mussels and use them either for as a product in the aquaculture in order to reduce the mortalities, or even to enhance the environment using the native mussels, but with more resistant against. Yeah, it's not exactly a genetical as its own. We don't have the information of maybe they might have an epigenetic, some epigenetic changes that could lead to these phenotypes. Here we have only, as we can say, the first layer of the answers. That's the response. And we are sure that they have better defense using higher energy in order to survive. In other publications, for example, we have mortalities that show that in 28 degrees, the non-hardened mussels will have 100% of mortality near 10 or 15 days. In difference, the hardened mussels have lower mortalities near the 50%. So we hope to have, that's exactly why we say equality. Okay, we might have a better, we would take a more enhanced organism, but we don't have the image of what changes can be happening in the mussel because our experiment is only for 20 days and 4 days of hardening process. But what happened in a period of 3 months or 4 months or even a year? Yeah. The cost. Okay, yeah. The cost is completely nothing if we take into consideration that we don't use any chemical or the cost is only to install human power, of course, and install this system. We need only some tanks and, of course, some heaters, and the whole process is energy. And the other one is when we want to use them in aquaculture, for example, there is also a cost of transferring from the aquaculture to the system. Of course, we can create a system that can be near the mussels. It's a system. It's also a system that we can easily install maybe in a boat. It's not. It's under construction. No, not needed. Thank you very much.
Κύριε Μπαντουβά, μ' ακούτε?
Ναι, καλησπέρα.
Καλησπέρα. Λοιπόν, σε μισή ώρα ξεκινάτε εσείς. Εγώ θα σας δώσω τα κατάλληλα δικαιώματα. Έτσι, θα κάνετε εσείς share την παρουσίασή σας. Εντάξει.
Πολύ ωραία. Ναι, είμαι συντεμένη.
Ωραία. Σε μισή ώρα, έτσι.
Απλά δεν έχω κακή επιστροφή ήχου. Σας ακούω με ηχό. Δεν ξέρω τώρα αυτό αν θα επηρεάσει την παρουσίαση.
Όχι. Είναι τα ηχεία που ακούγονται.
Μάλιστα.
Θα το κανονίσουμε αυτό.
Εντάξει.
Κύριε Μπαντουβά, με ακούτε?
Ναι, ναι, εδώ είμαι.
Λοιπόν, κάντε share την παρουσίαση. Έτσι. Μόλις έρθουν οι chairman, θα σας αναγγείλουν και θα ξεκινήσετε.
Θα ήταν καλό να ανοίξω και
Ναι.
κάμερα,
Δηλαδή
ίσως.
θα κάνετε, αν θέλετε, ανοίξτε την κάμερά σας, θα κάνετε share screen. Έτσι. Ωραία, εξαιρετικά. Ναι. Εντάξει. Λοιπόν, τώρα, οκ. Ναι. Μόλις σας αναγγείλουν, θα ξεκινήσετε.
Πολύ ωραία.
this session, Mr. Badubasa will present in online his work. It's about the age and growth of the squid Loligoforbesi in the Eastern Ionian Sea. Nicolo, you can start.
Thank you very much, Eugenia. Hello and good evening. My name is Nicholas Baduvas, and I'm very excited to present to you today in the university department I graduated from. Our work concerns the age estimation and somatic growth of the veined squid Loligoforbesi from samples that had been taken from the Eastern Ionian Sea. So let's get right to it. Okay. So to start with, the veined squid is a demersal squid species and one of the 4 native loligenids of the creek waters and the Eastern Mediterranean Sea in general, alongside the 2 Alloteuthis species and the common squid. It is also one of the 2 loligenids with notable commercial value in European waters, together with the common squid again, but their fishing landings are not routinely recorded separately, thus inhibiting both species fishery production and stock assessment. Its distribution range extends over the continental shelf of northern European waters, the Azorian archipelago to the west, and all throughout the Mediterranean Sea. In Greece, is considered to be of minor commercial importance, although we can't rightly know, as its landings, as we mentioned, are not recorded separately from its sympatric Loligobulgaris, and is of course mainly fished by bottom trawlers, as it's a demersal species. According to several studies, individuals can reach a maximum of 15 to 16 months of age, with most specimens not exceeding 1 year. Its biology, of course, is still poorly understood in the Mediterranean, and age estimations have been very recent. So our main objectives for this current work were first to estimate the age of our Loligoforbesi specimens coming from the Eastern Ionian Sea, use these measurements in order to describe our specimen's somatic growth rates, and finally to investigate any induced variations by sex and our distinct 2 sampling periods. So our study area was the Eastern Ionian Sea and its deep inlet, the Corinthian Gulf. Our studylith specimens had already been removed in the framework of 4 research and monitoring programs that had been previously undertaken by the Institute of Marine Biological Resources of the Hellenic Center for Marine Research, and they dated from 2000 to 2001 for our period A, and from 2014 to 2018 for our period B. So we processed our stored studyliths in the laboratory in order to reveal their microstructure. We mounted our specimens on glass microscope slides using crystal bond resin as to securely grind them on both their frontal surfaces. We ground and polished them using 3 waterproof sandpapers with successively finer grain size while they were submerged in water. And in order to ensure the visibility of growth microincrements, we monitored the whole procedure under a stereoscope and a microscope, which were both connected to an image analysis software. So you may ask, what are cephalopod statoliths? Much like fish otoliths, they are hard structures composed of aragonitic crystals, and they are located inside adjacent cavities of the cephalopod's cartilaginous skull, which is called a statocyst. You can see the pair while it is still inside the statocyst, and this is the actual size of the statolith. So you can imagine how hard it is to grind it using sandpapers. So this is the pair of the statolith as it has been removed from its statocyst. And after grinding, this is the desirable product. And this is the statolith structure on which we counted the microincrements on. This is the rostrum of the statolith, which is the structure with the highest growth rate, which renders the growth rings more easily visible. So having the microincrement measurements and knowing the age of our specimens, we needed to correlate that information with the dorsal mantle length of our squids. So we had to select among various growth equations that are generally used to describe growth in loligenids, and especially our species. And we did that by applying the Akaiki's information criterion, which is an equation measuring information loss in models in comparison to actual measurements. And in particular, we used practical correction of the criterion, which is applied for small sample sets. So variation induction was assessed in a similar way by estimating the difference in its subgroup's regression criterion value. We first estimated the criterion value assuming equal regression parameters for each covariate, and then we recalculated inserting values as they values for the parameters of the equation as they resulted by an analysis of covariance. This difference in between those values would give us an indication of induced variation, and as this value grew, so did the induced variation. And finally, we estimated the daily and instantaneous growth rates with equations of Forsyth and Van Hoekelen with a time interval of 30 days, practically a month. So out of the 155 processed statoliths, 117 were able to provide satisfactory growth increment readings after grinding. Our minimum squid age for both sexes and both periods was estimated at 137 days, and our maximum at 298 for a dorsal mantle length range from 60 to 386 millimeters. The model with the least information loss in describing the relationship between the squid's dorsal mantle length and age was the multiplicative for all cases, for all our subgroups, with the exponential equation giving a close second result. Correlation coefficients were high for all of our investigated squid subgroups, and growth was much higher in male specimens for both periods, as indicated by the slope of our model equations. Variation testing indicated significant differences again for our male and female specimens for its sampling period, but differences between the two sampling periods were low, as indicated by the significantly lower delta value. All this is evident in both our growth rate curves and graphs, with female squid curves, growth curves being below, and male growth curves being upper in the graphs, there seems to be an increase in growth rates between the two sampling periods for our male, something that we did not observe for our female specimens. Now, age estimations for our Loligophorbaceae specimens from the Eastern Ionian Sea exhibited shorter lifespans compared to findings from relevant studies from the Northeast Atlantic, but also from age estimations from the Western Mediterranean, although our sample mantle length range was also a bit shorter. And to conclude our findings in a few important key points, we observed the sexual somatic dimorphism that has been reported for loliginids, with males demonstrating higher daily growth rates than females, a fact that could be attributed to the energy diversion towards reproductive growth of female squid as they grow in age. And as we saw in the previous slide, and although there seems to be this increasing trend in growth for male squid, the two sampling periods during which our specimens were sampled did not show so prominent statistical differences regarding the growth of the dorsal mantle length, at least not at this point of time. And that is possibly due to the relatively stable environmental conditions that prevail in the deep waters that our species inhabits. So that's it from me. Thank you very much for listening. I don't know if you have any questions. I don't know if
Thank
anyone...
you very much.
Okay. Now I heard you.
Good evening. My name is Theodore Zoulias. I'm from Hellenic Center of Marine Research. And my presentation is about the trends of the fishing yield and ecological indicators of the Avragikos Gulf lagoons, and especially for Tsopeli, Mazoma, Vati, and Pogonitsa lagoons that are the main lagoonal systems in Preveza Prefecture, situated at the west part of Avragikos Gulf. What is the aim of this presentation? The aim of this presentation is to assess if there is long-term changes in environmental status and in fisheries management in relation to fisheries production by analyzing the fishing yield and the mean trophic level and fisheries imbalance indices for the period 1918 to 2020. How that can be done? Time series analysis. We use the fishing yield to estimate landings variations, the mean trophic level to estimate systemic structural variations, the fisheries imbalance index to estimate fishing pressure and carrier capacity changes. For the data analysis, the annual fisheries landing data converted to yield in meaning tons per square kilometers for these 40 years of time series. Now, the mean trophic level. The mean trophic level is a level which combines the landings of each species and his individual trophic value in order to see the relation between the yields and the trophic level. The individual values of each caught species or family species is seen in this table. The fishery imbalance index is used to assess if a fishery is balanced in ecological terms. The fishery imbalance index is estimated for each year, having the first year of the time series as a baseline, and it takes into account both the landings and the mean trophic level, which are combined in the following final expression. Statistics, one-way ANOVA analysis was employed to test significant differences among the lagoons in mean annual fishing yield, mean trophic level, and fishery imbalance values over the study period, as well as in linear regression trend lines at the 95% level. And taking pairwise comparison post-hoc test was also further employed for statistical comparisons following one-way ANOVA. Results. The average yield, mean trophic level, and fisheries imbalance indicators for the period of time series are summarized in the following table. As you can see, the yield average values, significant differences were observed only between the Vathi and the Pogonitsa lagoons. In the Pogonitsa lagoon, the mean trophic level value 3.37 was found to be statistically higher among the other systems due to the smaller percentage of Mucilidia species in the fisheries production, indicating the marine character of the lagoon. For the fisheries imbalance index, the values exhibited negative, the fishery imbalance index exhibited negative values, but there is no significant differences among the lagoons. Now, the trends, now about the trends. As you see, in all the systems, in Vathi, in Tsopeli, in Pogonitsa, and in Mazova, there is a statistical significant decreasing trend in the fishing yield. Additionally, there is a statistically upward trend for the mean trophic level, here, here, and here. And the combination of these two indices reflects that the system has a function and structure modification. On the other hand, for the fisheries imbalance index, for Vathi and Tsopeli and Pogonitsa, there is also a significant decreasing trend. And there is a negative values in most of the time series years. On the other hand, in Mazoma, statistical analysis showed that the trend is stable. So for the referred three lagoons, fishery balance index reflects a decrease of fishery resources, while in Mazoma, there is a balanced exploitation. Just also want to say that the upward trend in the mean trophic levels reflect the fishing up food web effect, and that the maximum values of the mean trophic level were recorded in the last decade of the time series. Now, a scatter plot of the yield versus mean trophic level was developed in order to observe the relationships between yield and the mean trophic level. From the scatter plots, we can see
that two groups are identified. The first group consisted of Bathi and Pogonitsa lagoons, where two subperiods are answered. In Bathi Lagoon, the first subperiod is characterized by high yield values in the low midtrophic level range values, where in the second subperiod, lower yield values as well as higher midtrophic level values with a wider range of fluctuations are clearly found. In Pogonitsa Lagoon, there is a clear separation of the last decade in terms of lower fishing production and higher trophic level. On the other hand, three common sub-periods are observed in the adjacent lagoons Tsopeli and Mazoma, but with different characteristics. The first sub-period has a signature marked by high yield values and wide range of the trophic level, especially in Tsopeli Lagoon. In the second sub-period, the trophic level values range continued to be wide, but the yield values are reduced. In the third sub-period, the yield values continue to decrease in both lagoons, while the mean trophic level range values pattern is different between the two systems. The main point of these patterns is that despite the fact that the systems are situated very close each other and belongs almost to the same watershed, there is a different, a significant variation in the patterns of the plot. That means that serious local anthropogenic impacts in the watershed, in the Gulf, in Avragikos Gulf, and in the lagoons, have caused gradually habitat and hydraulic alterations within the lagoons, different each one, altering their abiotic parameters and possibly reducing the requirement for the fish to entering into the system. Now, based on the trend line for the plot, we can assume that the study systems are heading from a period of increased fisheries production within their current capacity to a shrinking fish activity and to a new environmental balance of reduced capacity. The main difference lies in the fact that for Tsopeli and Mazoma, the reported changes were exhibited in 3 consecutive phases, in Bathi Lagoon in 2 balanced phases, and in Pogonitsa Lagoon, the transition was completed in the last decade of the time series. And conclusions? The fishing yield showed a clear decreasing trend in all the study systems throughout the study period. The mean trophic level index showed a significant upward trend in all the study systems, indicating a fishing up food web state. Maximum values were recorded in the period 2010-2020. The fisheries balance index exhibited negative values in most of the time series period for all the study systems and showed a significant downward trend in Bathi, Pogonitsa, and Tsopeli lagoons. The trends in yield production and the ecological indices demonstrate that Bathi, Pogonitsa, and Tsopeli lagoons are associated with a function structure modification accompanied by both reduced fishing effort or the productivity of the systems. In the Mazoma lagoon, it seems that the yield and the trophic level changes are matched. The serious local anthropogenic impacts in the watershed, in the Avragikos Gulf, and in the lagoons caused gradually habitat, hydraulic, and abiotic parameters alterations. The scatter plot for the yield versus the mean trophic level of each lagoon showed that the systems in question are hitting from a period of increased fisheries production within their current capacity to a shrinking fishing activity and to a new environmental balance of reduced capacity. Thank you.
Yes. Can we describe the situation in Mazoma and duplicate this to the other lagoons to change their management to improve
as it first
management because the abiotic and anthropogenic cannot be answered anymore, but at least use the rate of distraction.
at the first glance, we believe that there is proportional shift between landings and the mean level in this particular system. So that's the difference between among the other studied systems. That's the first explanation that I have to tell you. According to the landings, to the fisheries composition landings, it seems that it's a bit different from the other systems. I can say that for the Tsopeli lagoons, that the productivity in Tsopeli lagoon is characterized by the chlorophyll A concentration, and for Mazuma by the ammonium concentration. So this has a connection with the abiotic parameters. Excuse me? Yes, but it's in a very, very low yield, very low landings. But as it is recorded, I also... Yes, that's the table covers species for all the study systems. Okay. All right.
Hello, everybody. Before starting, I just want to explain that this is not my special subject. I was a fishery engineer in fishing technology, and I just catch. Okay. And excuse me because I will read mostly the presentation because that I told you this is my friend's presentation. And the feeding and feeding habits of fish vary from species from season to season. They can even vary within the species between different age groups and sexes due to changes in the composition of the prey. And we searched two Lepidotrygla species. One is the spiny garnet Lepidotrygla diosedei, and the other one is large-scaled Garnet Lepidotrygla caviglione. And both species have limited commercial value in Croatian fishery and are most represented as discarded species. The study of discarded species, especially in oligotrophic seas such as Adriatic, is important for their role in the energy flow, as they are usually secondary consumers of invertebrates. And you can see the data collected from commercial bottom trawls. And samplings was carried out in the
Southern Adriatic Sea, spiny gurnards were caught 8 nautical miles from the coast at the depth of 170 to 230 meters, while large-scale gurnards were caught 1.5 nautical miles from the coast and at the depth of 120 between 150 meters. And this area is characterized by muddy bottom, which also covers the seabed of almost entire southern Adriatic, and it's typical of areas with little water movements. And the stomachs of 30 randomly selected individuals of each species were sampled monthly to analyze their diet. The stomach contents were analyzed under a sterile microscope to determine the qualitative and quantitative composition of the prey. A total of 360 stomachs of spiny gurnards and 360 stomachs of large-scale gurnards were analyzed. And you can see the methods which was used in the study. And for results and discussion, we analyzed 360 individuals for each species, and you can see the male sex ratio of them. And acuity index and fullness coefficients are the graphics of them are given in the slide. In this study, the lowest feeding intensity, the highest proportion of empty stomach was recorded in the autumn for both species, while the lowest proportion was recorded in spring. Also, in autumn, misses were found as occasional prey, while in spring, they were essential for food item. In Greek waters, an increase in the number of empty stomachs of large gurnards has been recorded in winter. For spiny gurnards, you can see the prey composition of the stomachs and the percentages of them. Then the most higher, the qualitatively higher stomach ingredients was in spring, as you see for this species. And in the South Adriatic, in the depths inhabited by the spiny gurnard and large-scale gurnard, the most decomposed are species from the family Paleomenidae, with Palepenos langurostoris being the predominant species. In the Adriatic, this species is characteristic of bottle mud biosenses, with its abundance decreasing from south to north. And the highest catches of this species in southern Adriatic are achieved at depths between 150 and 350 meters. About large-scale gurnards, you can see the composition of the stomach, and you can see the species, Mrs. species especially. And conversely, the winter samplings were much more vulnerable stomach ingredients than the other Lepidopteryglia species. Yes, for discussion in other studies, species from the order Mysid are also cited as essential prey for large-scale gurnard. Emphasize that species from the orders Mysid and Amphipoda are the main food resources for large-scale gurnard in Greece. Terras investigated the diet in the Eastern Mediterranean during May and September and found that prey from other Mysis was the main food source, while the decapod crustaceans and other prey found did not contribute significantly to the diet. And also, sampling was only carried in daylight, and it was not possible to analyze feeding intensity around the clock. The difference in prey abundance and composition could be better explained by knowing the times when the fish were more active. Yes, this is the most important detail, I think, for diet studies. However, due to the different bathymetric distribution, they have different niches, allowing them to consist without direct competition. And this was a quick presentation. Thank you for your attention. And if you have any question, please write me and I will send it to Tatiana and maybe she will answer you because I'm not really talented about this subject. Thank you. Good afternoon to everybody. I wasn't supposed to do this presentation, but I was betrayed by my team. So basically, this is in the framework of a research project. We had the idea to get some blood samples from wild European hake and basically to analyze the blood samples. One idea was basically what we were going to find in the blood, and the other one, if we would be able to do some machine learning to decide whether we can find from blood composition some other things. Both are going to be published. This is the first part. So European hake is a species with high commercial value. It's overfished basically, and our idea was to get blood. It was quite difficult to get blood from live fish, because the sampling happened using an otter troll, and we had to do it very fast to get the fish get the blood where the fish were alive. And in general, blood sampling is used from fishery, not so much from fisheries, from aquaculture, basically to assess the general health and well-being of the fish. And we basically wanted to have an idea, because the most recent data on wild merluccius were back in the 70s on hematological data. So nobody else has done this up to today. So we decided, what the hell, you know? So we wanted to check if there is a change in the blood, the composition basically of the blood in time and based on different depths or different times. There is a small error here. We had 4 holes between 80 and 100 meters, not 8. Okay. And the holes lasted between 30 and 90 minutes each. It was a common otter troll. It was a commercial troll, which we basically rented for experimental fishing. We drew blood from 40 individuals using syringes and heparinized needles. And we assessed hematological properties. There are basically 2 ways to go about it. One is to do it manually. The other one is using a hematology analyzer. Usually they do not employ a hematology analyzer when you're doing blood analysis for fish. Our hematology analyzer, a colleague
did the analysis and was basically set up to do the analysis for fish. And what we found was that among all different properties, we found significant correlation apart from the thrombocytes. And our results, basically, we did one sample t-test comparison. They were compared to Argentina. We had the higher values, blood cells, the hemoglobin concentration in our results exhibited higher values than Argentina and Denmark. The last sampling that happened for wild hake was in Denmark, and hematocrit as well exhibited higher values than Argentina and Denmark. And regarding the troll depth and the troll time, what we found was that they both significantly affect hematological values. So basically, when you say that we drew blood from a single species, it's very important to say how long you've been doing the fishing and how long, from which depth did you get the fish. And we saw that individuals that were caught deeper than 80 meters had significantly higher values in red blood cells, white blood, hemoglobin, and hematocrit concentration. And the same the individuals that were caught from trolls, from the 30-minute troll compared to the 45, 60, and 90-minute trolls, exhibited higher values, again in the same variables. So basically what we came up with was that sampling depth and sampling duration both have a significant effect on the hematological parameters of European hake, which can be influenced by environmental conditions, sampling technique, hematological analysis method, pathological agents, sex, size, weight, age, and species. So there are many. There are many possibilities, basically, to alter hematological parameters in a fish, and you have to be very. You can provide unique data concerning the health status of the fish if you use this correctly, but you have to be very thorough and to pay attention. And someone that is doing it, the person who is doing it has to have a lot of experience, because data tend to change if you do it in a different way. So, and you have to account for all these parameters. And thank you for your attention. What is this? You like this one? Second one. Okay. This one? Yes. I'm not sure. I did not make. Let's show you a picture. It's not. I'll shoot my students. Yeah. No, this was just the idea was basically to check whether we can predict age and sex from blood parameters, which basically makes no sense for using machine learning methodologies, artificial intelligence. Okay, so we did that, so we can find, but it's much more difficult check the blood than to go and open the fish and see what is the sex. So there's no point. It was just something we try to do if it's possible. So the idea basically is maybe in the future we could create a database with different blood hematology parameters from fish from different areas of the world, from different depths with different health conditions maybe, so that we could more or less refer to this and assess the health of wild populations. So this would be a nice thing to happen. But everybody is fighting which is the best way to take blood, or which is the best way to analyze blood. So unless we have a common protocol, it's not going to work. But it's an idea. I'm hopeful. The problem is with the automatic hematology analyzer, because the blood cells of fish are not the same like yes. So there's a problem. No, not in Greece. In Denmark and in 60s and 70s, those two, Argentina and Denmark. So this was the last reference we could find. So, and we basically decided to compare ours with theirs to see what was the difference. But there are many possibilities to change the hematological parameters. So who knows what they did, how they got it? But it was an idea. Yeah, exactly. Thank you. Which one? No, there's no correlation with death here. This is correlation only with hematological properties. The depth was not a correlation. It was like a model. Here is the depth. Yes. We had 4 different depth variations: 30, 45, 65, and 90. I don't remember. By looking at this graph now, hemoglobin is here. Okay, so you check the depth. I think the depth is here. I think 95, 100. I don't have the exact numbers now. Okay, it's hemoglobin is this one, and this is the depth, and this is the troll time. But they change according. Yeah. Anyway. Depend, because the sampling we had sampling in the winter as well. So yeah, but yes. hemoglobin.
Because the depths are
Lower 80, 95, it goes less. It's less. Yeah, it's less. It's less. I can send you the raw data if you want.
And below this depth is the environmental conditions.
While we were analyzing the data, we missed in the beginning the troll depth and troll time. We thought there was no difference, and we used it as a uniform, basically. This is this. The problem we had, the major problem we have with this work, that in theory you cannot use an automatic blood analyzer. And so it was the first time. Exactly. I'm not a microbiologist, I'm a marine biologist. Any other questions?
Δηλαδή, αν μπήκα, ό,τι κάναμε σήμερα δεν μπορώ να το πούμε. Το YouTube live, μόνο. Τα προηγούμενα, δηλαδή αυτό που σώμα από 5 ώρες. Ό,τι λέμε για live το βιντεάκι, αυτούς σε δεύτερο χρόνο, το live, μπορείτε να μπείτε να το ξαναδείτε. Ή να το κατεβάσετε τοπικά, να κόψετε το κομμάτι που είναι το δικό σας. Αυτό όμως πρέπει να γίνει μέχρι σήμερα, γιατί αύριο θα είναι άλλο. Αύριο είναι άλλο. Θα πρέπει να κάνουμε σήμερα. Αλλά από ό,τι ξέρω θα πάει στην Ταζαφές. Μισό λεπτό.